All right. How we doing, everyone? Good. Sounds like it. Very convinced. All right. So we'll start with a little bit of introductions here. So this is my very busy slide, my visual CV of places that I've been, if you will. So I grew up in Lutz, Florida, from Tampa originally. Went to USF for undergrad and med school. Spent a lot of time at Moffitt. Then went up to Tufts for residency and Hemonc fellowship here and stayed on board. And this entire time, I've really had a complete and total focus on tumor immunology. I've really never wanted to do anything else since joining Cellus's lab at Moffitt back in oh God, how long ago was that? I don't think we want to talk about that. In 2006. So I mean, it's really a lifetime dedication. I really enjoy it, and I think it's a really cool time to be in medicine, particularly gastrointestinal cancers. Outside of this place, I hate ketchup, but I love travel and music and cars and not being.
So I'm Dan, everybody here knows me, I think, but I'm originally from Jefferson Hills, PA, which is just south of Pittsburgh. I did all of my schooling at the University of Pittsburgh, and then I came to Duke, and I matched here for residency. I've been in North Carolina ever since. My current role, as I'm sure all of you know, is cell therapy related on the malignant heme service, and so I really deal with all cell therapy applications, whether that be solid tumor or liquid tumor. And my primary interest, as you probably can gather, is related to T cell immunology. Other interests unrelated to science include travel, hiking, boating, and I have a twin who lives in California.
I didn't know that until I saw this slide.
Yeah.
I was like,
I don't
that's
advertise it.
No. We have a locket or something. All right, here's disclosures. Boring. Here's the outline. So this is meant to be really interactive. If we say something, you're like, what the heck is that? Or you didn't define that abbreviation, Nick, or like, Dan, I've never heard of that drug. Can you stop for a second? Please do that. There are no references on our slide. Everything on our slides, everything is in the Duke box. So if you want to do a deeper dive in any of these papers, you should go into the Duke box, read the paper yourself, email us with questions. We'll talk about it more. And then we're going to send you guys a feedback survey at the end, and we would really appreciate any thoughts on what you thought was missing, too confusing, what was really good, anything like that. We will accept it because this is the first time we're doing this lecture. And one of the reasons we're doing it is because I think if you talk to patients about how they think about the immune system, and I just searched boost your immune system on Google on the left, and that's what comes up. And so we think of this idea that we're going to turn the immune system into a tank that just kills all the cancer cells and then everything's fine. But it's not a tank. It's more like the Department of Defense, which is outlined in the top left. So if you don't understand how complex and interactive the immune system is and how it has something to do with everything else in your body, then how are you possibly going to manipulate it, deal with side effects, overcome mechanisms of immunotherapy resistance, sequence your treatment, etc. So the goal of this lecture is not for you to learn every single thing on every single slide. It is to learn how to learn tumor immunology, interpret some of these articles, and to I gotta get them out of the board. Yeah, sorry. Oh, sorry. It's okay. So we'll get it in post. Don't worry. Don't worry. And so what adds to this complexity is not only do you have multiple systems interacting with the immune system, you have multiple systems within the compartments of a tumor, and then you also have multiple different cell subtypes, their functions. How you should learn how we determine these. What are the impacts of therapeutics because these change emerging cell states. And what you see up there in the top right is single cell RNA-seq. So if you thought there was 2 types of dendritic cells, no, there's 6. If you thought there was 2 types of macrophages, no, there's 12. You can see the increasing complexity, and this is not going to get any more simple. The problem is that we are adrift in a sea of data, but we thirst for knowledge. We used to throw throw immune cells out of tumors when we would take them out of humans and mice to preserve specimens, which to anyone in this room, I hope sounds completely insane, but that's what they used to do. I was told I was wasting my time in a vaccinology lab. This is a complete dead end career. It was not considered important in oncology. Tumors were treated like infections, and I would argue still are in many ways, just like focus on kill the tumor. And we actually don't know the immune effects of like chemotherapy or targeted agents or a lot of the things that we use on an everyday basis. And this sort of like belies the point that immunology is really old and the concept of anti-tumor immunity is more than 100 years old. So we have this lack of foundational knowledge of tumor immunity, and then we get single cell RNA-seq results like you saw on the last slide. And we're supposed to say, well, okay, we know what's going on now. We're totally fine. And it's obviously not how it works. So I think there's a fundamental need to better understand immunology, to better understand tumor immunology. So why immunotherapy? So this is the 10-year follow-up of Ipi/Nivo in melanoma that was published earlier this year. And you can see that the median overall survival for melanoma-specific survival exceeds 120 months. So, I mean, just let's be honest here, like immunotherapy is the only thing that cures cancer because dacarbazine had a median overall survival of 9 months. And so fast forward today, these are two patients from my first line botensilimab balstilimab trial with microsatellite stable colon cancer. The patient on the bottom is 30 years old, has never experienced any type of chemotherapy. The patient on top had peritoneal carcinomatosis recurred after FOLFOX many years ago, and they're both responding and doing great, and they're getting only immunotherapy. So we're moving into a world where chemotherapy is going to start to take a back seat to immunotherapy. And so this is the changing plane of oncology, and we need to be at the forefront of it as learners and the next generation of researchers too. So I'm going to stop for a minute, and I'm going to do our first board talk. And again, like please stop me if something is too small or you don't see what I'm talking about or whatever. You just want to kind of like ask a question. So there is at the bottom is kind of a small rendition of like how a therapeutic cancer vaccine works, and I put that there because that's really like what got me interested in tumor immunology. Because when you vaccinate a patient, we know that you get antigen uptake in the lymph node, you get cross presentation, you get CD8 T cell migration to the site. So if you have a tumor should release antigens, and when they release antigens, they should be picked up by dendritic cells and other migratory antigen-presenting cells. But as we'll talk about, dendritic cells are the most important there. They then migrate to the lymph node. And in the lymph node, that DC should interact with CD4s and CD8s. And what's going on there? So the CD8 is binding through TCR MHC1 interactions, and the CD4 is binding through TCR MHC2 interactions. And all of this occurs because the dendritic cell was able to pick up this antigen, process it in a proteasome, cross-present it onto MHC1, and then make this migration back to the lymph node. And so once in the lymph node, once you have activation of what I like to refer to as the Holy Trinity here, you then get CD40 that binds to DCs. So we have your first signal here, and now we have our second signal. So CD40 binds to CD40 ligand on DCs. And in doing so, it upregulates co-stimulatory molecules, which we'll talk about, like CD80 and 86. And this binds to CD28 on CD8 T cells. And that's your second signal complete. Now, it hasn't even gotten to the tumor yet, or like tried to kill it. So there's a long way to go, right? So it goes to the tumor, has to migrate back, has to home to the tumor, has to be TCR specific, it has to recognize this antigen. And in doing so, what the CD8 T cell is going to do is it's going to produce interferon gamma. And it's going to produce perforins and granzymes. So this is the business end, is granzymes and perforins. They're what's actually killing the cell. And the interferon gamma is supposed to stimulate the tumor to be more essentially immunogenic. However, there's a very clear reciprocal here, which is PD-L1. And this is a normal tissue homeostatic mechanism that occurs in response to interferon gamma. And so what this means is that if you have PD-L1 expression in the tissue, that means you have interferon gamma production, ergo this entire process has occurred before that. So your PD-L1 staining implicates a lot. And it also implicates that there is interferon gamma in the microenvironment because this stimulates other cells like myeloid cells to produce PD-L1 as well, which we'll talk about. So this is essentially how antigen cross-presentation is working in the tumor, and how there's a normal mechanism of resistance that occurs that's just exploiting homeostasis. Of course, we know that like Pembro and everything else targets PD-1 and PD-L1. There's problems with that as well, like the existence of a PD-L2, which occurs primarily on myeloid cells. If you're targeting just PD-L1, you're missing PD-L2. We'll talk more about like the FC engagement there too. But the other thing that occurs in the lymph node here, so back even again independent of the tumor, is the upregulation of CTLA4. And CTLA4 occurs in response to TCR stimulation. And then it outcompetes, and this gets really tough to draw, it outcompetes CD28 because it binds at higher affinity to CD80 and 86, and it shuts down this response in the lymph node. So it shuts down response in the lymph node naturally, and shuts down response in the tumor in an antigen-specific manner. So this is why you have synergistic activity with things like ipilimumab and things like nivolumab, because they work in two different compartments of the immune system. And this is also why tumor vaccines don't work, because it's just not enough to just supply antigen. You have all of these other compensatory suppressive mechanisms that occur. Questions, thoughts on that board? We'll go back into it. So this is the kind of way we want to think about this, is that for every action, there's a reaction. There's always going to be a balance in the immune system, and tumors will always try to exploit that. So we have to think in terms of how do we manipulate the cancer immunity cycle. And so this is, again, this cancer immunity cycle, which is like one of those figures that's been in just like so many presentations that you're sick of seeing it, and then they added more stuff to it and more stuff to it. And vaccines fall here, where it's release of cancer antigen presentation into APCs. But you can see in green are all the what would be considered anti-tumor mechanisms, and in red would be considered pro-tumor mechanisms. So this is really just to show you a landscape of the balance. And if you understand that landscape, you can say, okay, well, this part of the cancer immunity cycle is missing, and that's why this and this would be sensible combinations. That's a grotesque oversimplification, but regardless, it shows you a little bit about how to approach this, and when you're thinking about where in tumor immunity that tumor is at that time. Because ultimately, cancers are an in situ neo-organ that represent a failure of both genetic checkpoints and then immune checkpoints. And I put Charles Darwin up here because this is the survival of the fittest occurring within our bodies every day. Your body has eliminated so many tumors that you just don't even know about it. Like, shout out to your T cells and DCs. But beside that point, we don't think of it this way. We don't think of it as this like organ that's growing and fighting with our immune system continuously, and then you give a therapeutic and you shift it one way or the other, and then the tumor tries to shift back. And this was conceptualized by Robert Schreiber. And if you were like, I only am going to read one paper from this crazy person's lecture, this is the one, because this is an awesome framework of how to understand tumor immunology. He described it as the 3 E's of immunoediting. And if you think of it as like this evolutionary struggle, kind of how I phrased it before, and how tumors can be eliminated, or they can reach a state of equilibrium, or they can escape. And that's when they become clinically present. And there's a multitude of mechanisms that lead to that occurrence. And we can give immunotherapies to push the tumor closer towards elimination and be hopefully aware of what it's going to do in reaction to that. This is really important too, because I think it's really easy for us to be reductive. And I can't tell you how many times I've heard very respectable investigators bring this up, is that mutational burden alone is not sufficient for tumor elimination and for immune recognition. And I thought this was a really elegant way of showing it. This is from the Tyler Jacks lab, where they knocked out MSH2, essentially making a KRAS P53 lung tumor microsatellite unstable, and it doesn't respond to checkpoint blockade. And the reason is because it does not have clonal neoantigenicity. It has too much antigen diversity, and so you cannot mount an effective immune response against the tumor. And if you look below, and these are really difficult samples to get, so I'll just, I will always emphasize that. This is all MSI high, colorectal, and gastric. And they looked at clonal neoantigen burden. And the fact that they have double digits of patients on this is like really magnificent. But you can see that if you have a subclonal to clonal neoantigen ratio that's low, you do much better. So it's better to have these clonal antigens that are recognized. And these antigens also need to be shared between TCRs of CD4s and CD8. So as we mentioned before, this process really only occurs effectively if the TCR and the CD8 and the TCR and the CD4 is the same, because where's the dendritic cell, the central component of that? So you're really only going to get this kind of specific recognition if you have TCR sharing, which I think is a concept we always liked, but we never really proved in this group that we put in a paper on with hepatocellular carcinoma describes that. So elimination, we'll start here because this is what happens every day. The innate immune system is the first line of defense against tumors. We'll talk about interferon signaling, NK cells, and CD40 signaling this part. I'm not going to spend a lot of time on interferon signaling because I feel like this is a pretty basic immunological concept. You could spend oodles of hours going through the different interferon pathways and how they've been manipulated and how they respond to different extracellular stimuli. But it's important to note that the main role of interferons, particularly in tumor immunology, is to increase the expression of CD80 and 86 on DCs. The problem is that tumors, and that gives you your second signal, obviously, like we were talking about before. The problem is that tumors often suppress JAK-STAT signaling, they suppress endogenous retroelements, and they also co-opt all of these pathways that you see at the top. So TLR4 can signal through NF-kappa B, and NF-kappa B promotes anti-apoptotic genes. So if you've got like lots of TLR4 ligands, which you think, oh, those will be immunogenic, you could actually be stimulating tumor growth because you're causing resistance to apoptosis. Tumors also rewire interferon signaling as more of a chronic mechanism, which we'll talk about more later. But I really want to get into NK cells here because I think this is more therapeutically applicable to this audience. These are short-lived innate lymphocytes that interact directly with several other immune cells. I think where they fall into this category here, if I'm going to add something to the board, it would be, well, that's okay, cool. It would be that NK cells are kind of hanging out here after they've recognized the tumor and they're producing FLT3 ligand and they're producing XCL1. And these are promoting these DCs to actually move to the tumor. So NK cells are kind of weird in a lot of ways. They're inhibited by MHC1, but they're also inhibited by HLA-E, and they're suppressed and undergo apoptosis in response to prostaglandin E2. They're capable of direct tumor killing. They're important in early tumor clearance, but tumors find many ways around NK cells. Strangely, there's a galectin-9 subset that is capable of pro-tumor functions. I sheerly mentioned that just to show you the dichotomy of every immune cell. So we'll talk about a few of these possible mechanisms, one of which is MICA A, MICA B shedding. So MICA AB is a pretty promising target. It gets cleaved off the surface of cells by proteolytic cleavage, and then NK cells essentially can't recognize it. So in tumors that have dropped their MHC1, of which there are many that decrease MHC1 or antigen processing, how do they survive NK cell killing? This is one of the ways, is that they cleave MICA A, MICA B, it's shed into the microenvironment, and NK cells never even reach the tumor, essentially. So if you stop that proteolytic cleavage, then you lead to NK cells carrying through anti-tumor attack. So as I mentioned, NK cells are pretty difficult to target for a number of reasons. Again, they're short-lived, so there's been lots of struggles with like adoptive cell therapy. But one really promising avenue is by targeting prostaglandin signaling, which we're going to talk about a lot throughout this lecture. But there's also other checkpoints and immune stimulatory receptors like NKG2D, which could come into play a little bit later on. Interestingly, PD-1 and PD-L1 do not lose activity when you deplete NK cell specific PD-1. But if you deplete NK cells in mice, it doesn't work. And the reason for that is because of this. They can't recruit type 1 dendritic cells without NK cells. But the activity of anti-PD-1 and anti-PD-L1 drugs is probably not directly dependent on NK cells. So that's something that's been brought up before, so I figured I would mention it. And again, we'll come back to PGE2, but I think MICA AB shedding is one to watch and to get a better understanding of as it starts to move into trials. There are so many other things that are going to target NK cells. If you really, really have an affinity for NK cells, I would recommend reading Dr. Rizvani's work. She has done amazing, amazing work at MD Anderson on adopted NK cell therapy. There are some recombinant IL-15 agonists that stimulate NK cells in vivo that could be really interesting, but these have toxicity problems. TGF-beta inhibition we'll definitely talk about, and then inhibitory receptor blockade, as I mentioned before, and then ultimately we would love to be able to give things like bispecific NK cell engagers or trispecific NK cell engagers that can then inhibit that MICA B shedding in the microenvironment, bring NK cells to the tumor, which then could lead to better DC recruitment. So there's a lot that could be done with NK cells in the near future. It's just a cell type that is not really commonly targeted right now, so something to be aware of. Now we're going to dive into the best cells, which are dendritic cells, obviously. So they orchestrate self versus non-self. There are well-delineated lineages. There are functional states that are being further defined. They require FLT3 ligand for development. And the way that I always remember this when I was learning immunology was that dendritic cells flit from one place to the other. So since they're migratory, I'm like, okay, well, they need FLT3 to migrate. So they also need XCL1, CCL5, CCL4. These are wimpy cells in culture. I'll be really honest with you. When you're growing them, if you like pipette them too much, then they die. So they're really frustrating to deal with and really hard to work with, like outside of the body, which makes them very hard to study. They, again, have these well-delineated lineages. People have made dendritic cells from fibroblasts by just inducing like 3 transcription factors, which is just bonkers. But we don't really understand their cell states very well, and we should, because they constitute less than 1% of immune cells. But if you delete just one cell type, type 1 CDC1s, those right there, if you delete them, you will not have any response to checkpoint blockade, and you will have tumor outgrowth. So we need to better understand these processes, and I would argue that one of the holy grails of oncology, and I've had at least 2 of my mentors say something to this effect too, is manipulating dendritic cells in situ. If you can manipulate these cells inside a patient without having to do any adoptive cell therapy or anything else, you're able to eliminate suppressive dendritic cells. You can push towards rejection of a tumor because that's what they decide. So there's a newer phenotype called MREG DCs that we'll talk about a little bit that the previous lab I was in did some work on, and there's some other papers that have come out that are really interesting. You can see, which is really, really nice. So even though I angrily say that they're hard to study, they are easy to study from this perspective, at least, is that they hold up between mice and human. And this is an important facet of tumor immunology that I will occasionally mention, because you'll go and you'll read a mouse study and you'll say, is this going to translate? And I think the most fundamental one, 2 of the fundamental ones that I saw recently is that PD-L1 has 60% homology between mouse and human, which is not great. And that all of our models that we've knocked out P53 in, well, mutant P53 actually has activity. And do patients have P53 knockout? No, they have mutant P53. And what does mutant P53? It upregulates Wnt ligands. What do Wnt ligands do? They suppress tumor immunity. So we're just missing this whole aspect of things because we're over-modeling and over-modeling and over-modeling. So take the mouse models with a grain of salt. They are useful, and they are useful in context where the subtypes of cells are conserved. So that's one thing that I just want to kind of convey there as somebody who has studied dendritic cells for far too long. And CD40 signaling, I call this the immunological white whale, because you'll read so many papers over the years that's like, we cured all the tumors in the mice because we gave CD40 agonism. Cancer's done, bye everyone. And then like that doesn't work in patients, and it's very difficult to know why. So as CDC1s are necessary, they're not sufficient. You have to have, again, this matched TCR diversity between CD4 and CD8 T cells, so you get co-stimulatory signaling. And CD40 ligand and CD40 signaling licenses APCs above the level of TLR agonists. And then these CDC1s can then be suppressed by MREG DCs in trans, by them interacting with the DCs themselves, or through the generation of T regulatory cells. So DCs, again, have this very critical balance between self and non-self. And we'll talk a little bit more about these cells as we go on. But you can see how important this pathway is, and the fact that these MREG DCs that are suppressive also express CD40 may be a reason why CD40 agonism has failed to come to fruition. But this was one of those, like, we think it's going to be really promising. And the only thing that we've really gotten out of CD40 signaling is that CTLA4 does enhance CD40 and CD40 ligand interactions. And that's probably because it's improving this TCR matching. When you have like more CTLA4 blockade, you have more co-stimulation in the lymph node, right? And that's where your helper T cells are. So if you have TCR specific helper T cells, then it's possible that that is what leads to enhanced CD40 ligand interaction. But this paper really nicely showed that you could like separate these regulatory DCs into like DC1s that promote anti-tumor immunity, or the most common DC in tumor types, in tumors, which is DC2s. So MREG DC2s then promote Tregs and then suppress anti-tumor immunity. So this like bifurcation and this observation that you have mostly DC2s in the tumor is, I think, really, really important to this like initial step of elimination. So MREG DCs are kind of like the final boss of tumor versus self. They're, again, metabolically distinct. They've been described in, I don't even know how many cancers. Michael in Brent's lab was able to find single cell RNA-seq data sets from at least 6 different tumor types where they were detectable. They may separate whether you're getting like CD4 specific, TH1 specific TCR or Treg TCR specific interaction. So it's really, again, that fulcrum of self versus non-self. And this is a really nice paper where they looked at HCC and they looked at immunotherapy response. And most of their responders had this triad of MREG DC1s, CXCL13 CD8s, which are migratory CD8 T cells, and then TH1s. And then the non-responders had Treg, MREG DC interactions. And they did all of this with spatial analysis, which we'll talk about a little bit later. So I think these cells are really important. They're really rare. They're really hard to study. And this is going to make them really important in targeting. Equilibrium, I'll just say briefly, there's a tumor in place, but it's at a stalemate. This occurs during dormancy, and it's relevant to treatment in the locally advanced and the long-term monitoring setting. Patient tumor heterogeneity plays a role, and I think the best way to think about this is in the terms of neutrophils, where cancers can be escorted by neutrophils, or neutrophils can kind of prep the soil so tumors grow better. Tumor-associated neutrophils are very short-lived. They're often termed myeloid-derived suppressor cells. Tumors can activate emergency granulopoiesis, producing the GCSF that we all have given a million times in clinic. Hope you sleep well at night. They also express CXCL1, 2, 5, and 8, which bind to CXCR2, and they promote PGE2, which suppresses NK cells and sustains neutrophils. These are, I think, if there's anything I ever saw in the lab that was more anti-correlated, I don't know what it was, but it's like even on the mouse-to-mouse level, you would see like higher levels of PMN MDSCs and lower levels of CD8s. It's a very strong association. And we know, too, that there are anti-tumor neutrophils, but that is not the predominant function here of neutrophils in cancer. And we know this because TAN-high tumors are seen across histologies, and they do much worse than patients who have fewer neutrophils in their tumors. They have fewer metastasis, they have better disease-free survival, and better responses to therapy. So neutrophils are really important. We'll talk a little bit about their targeting as we go through, but as to date, there is not a lot of effective neutrophil targeting, mostly because of toxicity. I think this is a promising mechanism, and I'm of course biased because this is pictures from my paper. So PMN MDSCs and NK cells both express EP2, EP4, which are immune suppressive PGE2 receptors. And this is a dichotomy because EP2, EP4 will prolong the survival and function of PMN MDSC, and it does the opposite in NK cells. So this is an XN5 staining here, and you have a reduction in apoptosis, and XN5 measures apoptosis. In PMN MDSCs, when you have a Gle2 active tumor that have lots of prostaglandins around, and if you block EP2, EP4 with TPSD 1495, then it restores apoptosis. And the opposite happens with NK cells. There's more dying NK cells in a high prostaglandin environment, and if you block EP2, EP4, then you are able to restore their survival. So I think this is a nice example of how tumors can manipulate kind of two arms of the immune system simultaneously. Escape is everything else we're going to talk about. So I'll stop there. Over to you. Oh, this is a great time to ask questions, man.
I'm really interested in this concept of like clonal versus subclonal antigens.
Yeah.
Antigens. What do you think is, so like all those tumors were MSI high, right?
Yeah.
Do you think that like the subclonal ones, like DNA repair got knocked out later in the tumor evolution process? They're all kind of like driven, right, by mismatch repair, but I guess why are some cancers have like a lot of clonal versus stuff? So what do you think is going on that's like driving the difference?
It's a good question. I mean, I think one of it is like the mechanism of DNA repair, like POL mutants in certain MSI patients will have like far more frame shift mutations, which create like higher antigenicity. So it's not just about like the quantity of the antigen or anything like that. That's about the quality. There's a really interesting paper that came out a week or two ago that was looking at low TMB responding patients.
Okay.
And that may be an avenue to find out. But I don't know anything on a genomic level that's been shown. So what you probably have to do is like single cell DNA-seq, after that. But I think it's a great question.
Because that's how we
ask that
figure
question.
out. Yeah. This is how we figure out who might respond, right? At least with the therapies we're looking at.
Yeah.
There's some way to like actually measure that with both data that we actually get.
You can, yeah, no, that's a really good point. You need to overlay it with like TCR sequencing. But we'll talk later, and what I'll do is make sure to remind me to point out that Tyler Jack's paper and gosh, I'm forgetting his name right now, but I'll send you guys other paper too. But these are great questions, man. That's a very good point.
Okay, so the first section that I have that I wanted to chat about is central and peripheral immunity. And I know this concept is fairly broad, and a lot of what I wanted to talk about today is related to therapeutic targets, potential therapeutic targets, specifically in the peripheral setting, but it's important to at least step through what central looks like as well. So central, I like to think of as the car wash, and peripheral is more so the detailing that occurs afterwards, where in the central immune system, right, you're eliminating the vast majority of your autoreactive cells. Peripheral is more so that fine-tuned cell subtypes that are being sorted through. So your primary organs for central immunity are going to be related to the generation and the testing of immune cells, right? So your bone marrow for hematopoiesis and B cell maturation. The maturation is maybe a little bit too strongly worded there, because you really are shooting out immature cells until they get out of the marrow. And then thymus, of course, for the T cell development process to occur and the selection of T cells. So I think there's three main concepts that are important, especially when we think about biomarkers in this setting. Number one is clonal deletion. It's probably number 1, 2, and 3 realistically, but it wouldn't be enough to just talk about deletion. So clonal deletion is a process by which T cells undergo negative selection, but they also undergo positive selection in the thymus. The positive selection portion is really just a testing to make sure that the MHC on the target cell and the T cell receptor are functioning effectively. There's some co-stimulatory ligand association here as well, making sure that CD4 and CD8 are functioning appropriately, but really, provided the T cell receptor is able to bind adequately to the MHC, then you're going to have positive selection. That cell is going to be moved forward in the process. Negative selection is probably more important, because this is when T cells bind too strongly to an MHC. And the reason this is important is in the thymus, it's binding too strongly to a self-antigen, right? So these are the cells that want to be eliminated. This is negative selection. These cells undergo apoptosis. Interestingly, though, positive selection for cells that don't make it through also undergo apoptosis, but the pathway is different. So positive selection you typically would see being associated with FOS and its ligand pathway, whereas you see AIR, or this autoimmune regulator, plays more of a role in negative selection of T cells. I'll be remiss if I didn't mention B cell maturation, but all I'll say about it is that it has a lot more to do with somatic recombination in the marrow than it does anything else. A lot of what happens with B cells in terms of class switching and all of that is what happens in the secondary, tertiary lymphoid organs, like your lymph nodes. Energy, also something important to talk about. This is a co-stimulatory process, and it's a co-stimulatory process in that you could have adequate binding of the T cell receptor to the major histocompatibility complex all day long, but if you don't have appropriate co-stimulation or you have downregulation of co-stimulation, you're going to put your T cell into energy. And energy is basically this non-functional state, but it is explicitly different from exhaustion, which we'll talk about in just a little bit. And these are both important topics when we think about, everything on this slide is related to how do we prevent the immune system from allowing autoimmunity. But what cancer cells are able to do and what tumors are able to do is leverage every single one of these systems to subvert our immune system, right? So when we talk about clonal energy, this is something that could be good for a functioning immune system, but absolutely in the case of tumors, leveraging energy is a bad thing, right? Because we don't have functioning T cells or B cells. So some example disorders, I won't say a lot about this, but SCID and thymic aplasia are probably the big two when we think about central. SCID is only relevant to talk about in this setting because of the fact that mice with SCID models are very valuable because they can be xenografted with essentially anything you would like to put in there, and you can test tumor types and biologic responses of different PD-1 inhibitors, for example. So
you can humanize the mouse model, something like human bone marrow.
Yep, exactly. And then thymic aplasia, this is primarily a disease of the pediatric population where peds basically don't develop an appropriate thymus, and you can see underrepresentation of different clonalities of T cells in those pediatric populations. Thymus transplant is really the only way around that. But I think it's important to note that as time goes on, everybody develops thymic aplasia, right? So by the time you get old, you really have no functioning thymus. And so the importance of developing appropriate T cell clonality at a young age is extremely important. All right, so let's hop forward here. So this is going to be very basic to start off with, but I am going to build on this as we go throughout this section. And Nick hit on a lot of what I'm going to diagram here already. So I'm just going to reinforce it in the first part here. So this is basically showing, I'm really just going to give the example of a T cell and what it looks like when it interacts with an antigen presenting cell, and we'll do a dendritic cell because Nick loves those. Okay, so this doesn't have to happen anywhere specifically. We're giving the example of central, but this is probably more so a peripheral process that we're diagramming here because in the central, you're not necessarily interacting with traditional antigen presenting cells. I'll say that. Okay, so you have your TCR here on the surface of the T cell. And it's important, first off, that it's able to bind to the major histocompatibility complex, right? So this could be a type 1 or a type 2 major histocompatibility complex. And the MHC type determines what type of T cell it binds to, right? So you have your CD4 and your CD8s. Appropriate interaction here is the number one portion of the process that's important. But of course, number two, and equally important, if not more important, is co-stimulatory ligands. So in the case of this, you need to see binding of B7, which is also known as CD8086, to CD28 on the surface of a T cell. Call this B7, but just know that it can be CD8086. If you have appropriate binding of these two here, then you have co-stimulation and the T cell can be activated. If you do not have appropriate stimulation here, or if you have inhibition from other ligands binding, then you do not have appropriate activation of the T cell. And so this dysfunction that you're seeing right here is what we just talked about in terms of energy. So you can have all the T cell receptor binding to the MHC you want, but if you do not have co-stimulatory activation, then you would not have activation of the T cell. So this is one of the major mechanisms by which the tumor cells are able to subvert T cells, because while this diagram is a dendritic cell, an antigen presenting cell, this can just as easily be a tumor cell. Any questions about diagram? So T cell subtypes. This has been hit on a little bit from what Nick talked about, and I alluded to this already when I was diagramming, but you have two major T cell subtypes. The MHC that they bind to is how I've separated them on the slide. You have your CD4 positive, which are your general T helper cells, and you have your CD8 positive, which are your cytotoxic cells, right? So these are your killers. Some important subtypes to talk about from a T cell subtype standpoint are your Th1, Th2, and Th17, and then we'll talk about Tregs as well. Th1 helper cells are what I think of as your classic T helper cells. These are where you recruit your cytotoxic response. So they're going to bring not only your CD8 positive cells, but they're going to bring your macrophages and other pathogen defense immunofighting cells. And they are notable for releasing interferon gamma and interferon alpha, which Nick talked a little bit about gamma, and we'll talk about that more as we go through. Th2 cells, I think of these as being on the opposite end. So these are your humoral immunity cells. So these bring B cells to the site of action. These are the ones that are linked typically with allergic responses. But the important part to note about Th2 cells is they're linked with a pro-tumorigenic immune profile. And it's really IL-4 and you could argue IL-13 that are associated here. And it's IL-4 primarily that's associated with tumor-associated macrophages that can worsen immune profiles. So too much Th2 in certain circumstances is not necessarily a good thing. Okay, and Th17, this is fairly recently described T cell subtype. The only important part to point out here is the differentiation ability of Th17 into Tregs or inflammatory Th17 cells. And the reason this is important is you always have this fulcrum, this balance of inflammation versus immunosuppression happening in your immune system. And if something, for example, a cell signaler were to push too much Th17 to Tregs, then you have the ability for cancer to subvert the immune system, right? Because you have too much immunosuppression in that circumstance.
And in the circumstance that they go towards inflammatory Th17, this has been associated with immune-related colitis. So they're really pretty problematic cells.
Okay, regulatory T cells, alluded to that already. These are your classically immunosuppressive T cells. They secrete things like IL-10 and TGF-beta. Those are two of the bigger immunosuppressive signaling molecules. But the other thing that they can do is they express CTLA-4. And Nick talked about this a little bit already, and we know that that's a druggable target right now, and we'll talk about where that actually fits into the picture over here in just a minute. But just know that they constitutively express CTLA-4, whereas your other T cell subtypes can produce CTLA-4 on their surface, but it has to be upregulated. Tregs automatically have it across the board. You'll hear regulatory T cells also referred to as CD25 positive, CD4 positive T cells. Okay, so CD8 positive, we already talked about the general premise of a CD8 positive T cell, but there's a specific T cell subtype that's important to talk about, which is this TCF1 positive. And there's a couple reasons this is important. It's a T cell subtype that has high stemness. It's like a progenitor CD8 positive, or it's like a progenitor CD8 positive T cell, and that's important for a couple reasons. Number one, it's associated with high persistence. And so we know in CAR T that higher rates of TCF1 positive, CD8 positive T cells are associated with better persistence and better response. What we also know is that they're correlated with response to checkpoint inhibitors. So they tend to have high PD-1 associated with them. Okay.
What is TCF? Is it
It's a transcription
factor.
Yeah. It's weird.
Or activation?
It's a transcription factor that's like determinative of lineage. So when they differentiate into either T-PEX cells or T progenitor exhausted cells, they express TCF1, which confusingly is encoded by the gene TCF7, because why not? It's really fun. And then, or they can be terminally exhausted with this tox. Yeah. John Weir is the author.
Yeah, I'll talk a little bit more about tox here in a bit. Okay, peripheral immunity. So again, this is the detailing. This is the secondary, tertiary, and quaternary lymphoid organs, but primarily the lymph nodes, spleen, the MALT, is where you think about for peripheral immunity. And this is really preventing of autoimmunity on a smaller cellular level. Preventing autoreactive lymphocytes, though, in the end is the goal. So a lot of the processes that are general premises used by the peripheral immune system are very similar to the central immune system for regulating autoreactivity. Tregs play a big role here in the peripheral immune system. And you have to remember that Tregs, in terms of targets, are very much driven by FOXP3. This is kind of seen as a master regulator, as a transcription factor associated with regulatory T cells. And I kind of have immune privilege as a sub-bullet here associated with Tregs. But it's important to remember that the premise of immune privilege, which is basically sites that do not have as strong of an immune response as other sites in the body, like the CNS and the eyes, were originally thought to be just physically related, meaning that because there was difficulty of different types of cells getting into those sites, there was less immune response. So tight junctions are the classic example there. We know that it's harder to get into the CNS. But what has been shown more recently, it's not just that, it's also cell signaling that downregulates immune response in some of those immune privileged areas. Clonal deletion, I won't say a whole lot more about that on the peripheral side because the process is very similar. There's an autoimmune reactive pathway that occurs if the cell were to have autoimmunity on the peripheral side as well. And energy induction looks very similar on the peripheral side, with the exception of the fact that we have a few unique biomarkers that play a much larger role in the peripheral immune system than they do in the central immune system. And the two big examples there, of course, that we've referenced ad nauseum and will continue to do so are CTLA-4 and PD-1. So here's the journey of a T cell from a naive T cell all the way to the terminal effector T cell or memory T cell. And I've inserted here, underlined some of the important ligands that are associated with each of these checkpoints. So you have your naive T cell, which we don't talk a whole lot about. We don't talk a lot about quiescence here, but quiescence is an important factor that can play a role in silencing of the immune system, especially when it comes to tumors. And they do this via the two big mechanisms I have down here, which are TGF-beta or TOB1. And quiescence is basically the silencing of the cell by keeping it in the G0 phase. And so the G0 phase basically means that the cell is metabolically inactive or has very low rates of metabolism, transcription, translation. But if it were to ever come out of quiescence, then it could mature normally. There would be no issues. And this is important because that's not the case with some of these other checkpoint areas. So to move along, prime T cell is the T cell that you see can undergo energy. Right, so a prime T cell means that the receptor has bound to the MHC effectively. There's no issues there. But again, lack of a co-stimulatory interaction or a weak co-stimulatory interaction or even inhibition can lead to something called energy. The important thing to remember about energy when you think about it versus exhaustion, because they can be confusing, is that energy occurs very quickly. All it takes is a single lack of co-stimulation to allow for energy, whereas exhaustion is notable for the fact that it occurs over a long period of time. It's the result of a long period of time. Two of the ligands that I have on here, LAG3 and CD73, associated with energy. It's important to note that PD-1, CTLA-4 can also be associated with energy too. But more notably associated with exhaustion. Exhaustion is a very hot topic for T cells because of the fact that it's such a big problem. We'll talk about that more as we go along, but exhaustion is basically the result of persistent antigenic stimulation over a long period of time without resultant effects. And so the big examples of that I'll talk about are cancer and viruses. And what you see as a result of that is changes in the T cell phenotype that lead to a phenotype that basically is ineffective. So I have a diagram here of all the big name ligands that interact with T cells. So you have your antigen presenting cell, and like I said, antigen presenting cell for our intensive purposes is always interchangeable with the tumor cell. And you can see the effects of all these different ligands on the T cell. So inhibition from PD-1 and CTLA-4, upregulation from your normal binding to CD28, as I have over there on the diagram. But it's important to know some of these big things. TIM3 is another big one. TIGIT is another one. Okay. let's hop back over to the board. Any questions so far about didactic stuff, boring stuff? Okay. So we're going to get a little bit more into checkpoint inhibitor interactions as we go further through this portion of the deck, but it's important to understand what it actually looks like when a checkpoint inhibitor interacts with a T cell and your antigen presenting cell. So as I mentioned, this over here is CD28. This is an important co-stimulatory molecule for binding to antigen presenting cells, and it typically would bind to 7. I'll write that down here. But in certain circumstances, we're able to see that that is not the case, right? So we know on certain types of T cells, you can see upregulation of things like CTLA4, or on T regulatory cells, you see constituent activity of CTLA4 on the surface. So what can actually happen is, as Nick mentioned earlier, if you were to have a T regulatory cell come into contact with a dendritic cell, for example, and this ligand is CTLA4, you have a very different interaction that occurs here. It's still an interaction, but what you're preventing from happening is the binding of CD28, which is probably still hanging out over here on the surface of the T cell, but it gets beat out by CTLA4. So CTLA4, based on the action that I showed on this previous slide, doesn't allow us to go back. But let's just say that
We're just going to move forward.
We're just going to move forward. There's no going back in life. CTLA4 is an inhibitory ligand, right? So you guys already know that, doesn't matter. And so what happens as a result is you see downregulation, right? CTLA4. So the premise of a checkpoint inhibitor is, can you block that from happening? And so what a checkpoint inhibitor ideally would come in and do is it would be a little Y-shaped antibody, and it would come in here and it would bind to CTLA4 and it would stop it from binding to the antigen presenting cell. T cell can live on and be happy and exert its cytotoxic effects or its helping effects or whatever it may do. And I'll come back up and reference this here in just a minute when we get into some more of the checkpoint inhibitor interactions.
Yeah, sorry. Are all of them, and you can't go back, but if we could imagine, like they were at lunch, so were those different inhibitory versus co-stimulatory, or what were the other ones that I?
Yeah. So yes, everything on that slide would be the equivalent of CTLA4 in terms of it's an inhibitory or co-stimulatory ligand. It's just we don't have drugs for all of them at this point.
And some are in development or have other issues like binding partners and things like that. So I think there's a lot. There's some really, really good immune checkpoint reviews out there that we posted.
Yep.
The immune checkpoint inhibitors are not playing a role in inhibiting exhaustion. They're just blocking the blockade.
Correct, for the most part, yeah. I mean, so something that we'll talk about is the importance of using checkpoint inhibitors early because of the fact that once a T cell is exhausted, it's very difficult for a checkpoint inhibitor or anything else to pull that T cell out of its exhaustion.
I don't really know if it's even possible. Like speaking of John Weary's work from before, he studied like tox reversal, if you can reverse terminally exhausted T cells. And this is like a real problem because chemotherapy generates tox expressing terminally exhausted CD8 T cells. When I mentioned before, it's like we don't know how chemotherapy affects the immune system as well. Maybe if you give it for too long, you're actually reducing cells. So, yeah, it's difficult, but I agree. Like the timing is hugely important.
And is energy in terms of its reversibility?
Is what different? Sorry.
Energy.
Energy can be reversed. Yes. It's easier to bring a cell out of energy than it is exhaustion.
Okay. So like the lactic inhibitors, they actually take like the energy T cells
and pull them out. Yeah. Yes.
Or do they just prevent the generation?
In which case did you say?
Like a lactic.
Oh.
For example, I mean, I guess maybe don't.
Dario Vignali knows. So Dario Vignali at Pitt has studied like LAG3 dynamics a lot, and they can actually co-associate homodimerize with PD-1 and stimulate it on the same T cell. So LAG3 is funky. It's really weird. It also like relatlimab, right? So relatlimab it binds to like the external portion of LAG3, But LAG3 can also has this like hook effect where it will bind to PD-L1 and PD-1 and strengthen its interaction with another cell, and it doesn't block that. So LAG3 is real finicky, and then like to the point of the terminal exhaustion, like those cells often express TIM3, but I mean, who knows if you can affect them.
But you should be able to pull a cell out of energy if you can actually allow that co-stimulatory process to occur, which ideally these are doing. Yeah, with that dimerization. Yeah. Not a whole lot to say here. I mean, cancer is the big taking point. Okay.
PTLA4, you get lupus. That's like the sum of that.
Yeah.
You delete PD-1, nothing happens. Like
Yes, right, correct. And it's all based on which cells these are found on, right? Which we're going to talk about here in just a second. So checkpoint inhibitors, while they are in some ways a magic bullet, they are not perfect. And we've alluded to this already, that mutational burden is important here. It's not the only thing, but it is important. And so mutational burden, new antigen burden, right, which is a surrogate MSI high, right, or tumor mutational burden. These are all buzzwords, and these are FDA approved related words. But really what it comes down to is the amount of mutational burden that tumor has. There is a general correlation with efficacy of your checkpoint inhibitors. So at lower mutational burden or higher amounts of variance that you see, there's almost a camouflage of those immune cells for checkpoint inhibitors. And so this obviously is a major problem. Additionally, downregulation of MHC1, right, so your CD8 binding MHC, is something that can be acquired, and it can be both genetic or epigenetic. And so there's been some preliminary studies looking at, can you affect this via hypomethylating? Hard to say at this point whether or not there's any promise to that, but we do know that some of this can be epigenetic. Okay, so we talked a little bit about exhaustion states already. Again, I mentioned that cancer and chronic viral infections are the big two because of that persistent antigenic stimulation. We know that sustained antigenic stimulation, specifically of your CD8 positive lymphocytes, is a serious problem. Again, it's resistance to checkpoint inhibitors. It's resistance to a lot. It's not just your checkpoint inhibitors. And it's dictated by this transcription factor 7, which we already talked a little bit about. More of this transcription factor 7 generally correlated with better response to immune checkpoint inhibitors. And again, talked about this already as well, but preventing it is much easier than getting a cell out of exhaustion. And tox manipulation is an important factor here. Nick talked a little bit about John Ware's work showing that you can actually ablate tox and potentially affect the downstream proliferation and differentiation potential of an exhausted T cell. But this has never really actually been shown in vivo, of course, right? So this is all just in vitro in John Ware's lab. Yeah. Anything to add about that?
No, just gotta watch our time.
Okay, yep. Oh, sorry, I'll run through. So this is inflammation over time. You can see the only other thing that I want to say about this on the slide is over time with this increase in inflammation and as the T cell moves towards the terminal exhaustive state is the upregulation of inhibitory ligands on its surface. You can see the massive collection of PD-1, CD160, LAG3 on the surface as the T cell experiences inflammation over a period of time, increased apoptotic potential, decreased ability to secrete things like interferon gamma. Antibody classes, everybody here knows everything on this slide, so I'm not going to go too much into this, but this is important when we talk about what checkpoint inhibitors do or can do, because of the fact that IgG can activate the complement cascade. Those are the only two things to worry about on this slide. We know that IgG antibodies are a single functional unit. They're Y-shaped. They have 2 heavy chains, 2 light chains. The potential for activity with activation of the complement cascade is a serious problem, right? So the Fc portion of your antibody is what could activate the complement cascade. So this portion down here, that's your Fc region of fraction crystallizable. This is something that historically was thought to not do a whole lot. But with the advent of monoclonal antibodies, bispecific antibodies as well, we know more and more about what this can actually do, positive and negative. And one of those is activate the complement cascade. So when you think about activation of the complement cascade, the downstream result of that is cell death, right? So if you bind an antibody to something and you pull NK cells in there and you kill that, well, you're going to kill whatever your bound cell was. So in the case of PD-1, which is a T cell surface cell marker. Do you want to kill T cells? Obviously not, right? So it's really important for these antibodies to be engineered or endogenously not activate the complement cascade when you're manipulating T cells. So in the case of PD-1 inhibition, like Nivo and Pembro, biggest two that we think of, these are IgG4 based and they actually don't have any strong Fc receptor interaction. So they do not pull in the complement cascade, which is important. PD-L1 inhibition. So PD-L1 we typically think of as being on tumors. This is bad, right? So again, we want to activate the complement cascade? Probably, right? At least theoretically it makes sense. That being said, you have to think about the fact that PD-1 and PD-L1 are found in your body whether or not you have cancer in there. And so where else is PD-L1 found? It's found on antigen-presenting cells and other important cells in the immune system. And so if you delete PD-L1, sure, you may kill some tumor cells, but what you're also doing is you're killing helpful cells. And so your big two PD-L1 inhibitors, atezolizumab and durvalumab, are IgG1 based, which innately would activate the complement cascade, but they've had engineering of their Fc domain to actually reduce complement cascade potential. Now, there is an exception to that rule, and that's avelumab, which is actually wild type IgG1, and it does activate the complement cascade. Okay, on the flip side of things, CTLA-4 inhibition, IPI is your biggest example here. Have to remember CTLA-4, the biggest hitter here is regulatory T cells. And so down regulation of regulatory T cells could be a good thing, could be a bad thing, because you also see CTLA-4 on other types of T cells, CD8 and other types of CD4s. But you see that IPI actually does activate the ADCCs. You're actually killing T cells, regulatory, and maybe others by doing so. Tremelimumab, on the other hand, IgG2 based, does not have ADCC activity. So when you're giving each of these checkpoint inhibitors, you have to be cognizant of the fact of, are you just inhibiting the target, whether that be PD-1, PD-L1, or CTLA-4, or actually causing downstream toxicity? We know that the Fc region causes a lot more toxicity than this, but we don't have to get into that today. That's the end of mine.
It's this while you switch.
All right. We're going to motor through these last sections so you guys can get to your activity of why drugs fail. Sorry, I put this on the slide just so I can use a great New Zealand accent. So microenvironment and spatial arrangements in the tumor are just like the hottest topic that's ever been right now, because we're starting to really appreciate that, as Dan mentioned several times, every cell is an antigen presenting cell, and where the disease is located is very determinant of outcomes in several different cases. So in the trial I showed you before, where we're using a second generation Fc modified CTLA-4 inhibitor, like botensilimab, or in our Vilastobart trials with Xilio, we don't have a lot of patients that respond that have liver metastasis. So it's very important to note that, and we don't recruit some of those patients for that reason. So there's a lot more to learn there. But there's also important interactions that occur on the tumor microenvironment levels. So keep in mind, when we biopsy a patient's tumor, it's a snapshot of both time and location. Tumors are heterogeneous by site of biopsy and the location. And so bulk methods of sequencing miss all of this. So this is from the TRACER-X study that Charles Swanton did in the UK, looking at non-small cell lung cancer patients in early stages and the association of immune cells in those non-small cell lung cancers. And by arranging which immune cells are in the stroma and which immune cells are in the tumor, you're able to predict phenotypic subtypes of different non-small cell lung cancers, and it's correlated to things like recurrence rate, smoking status, etc. So you get a lot more information knowing where your cells are, because these interactions with each other are so unbelievably important. So how can you do this? This is an example of mass cytometry, which uses mass spec on different slides. There's also multiplex IHC, and I'll show you various slides that include spatial transcriptomics, or something called site-seq, which is a way of detecting protein using RNA sequencing. And again, this is important because under normoxia and normal oxygen content, you do not have a bunch of cancer-associated fibroblasts or tumor-associated macrophages moving in. So you have a low amount of TGF-beta, MMPs, lysyl oxygenase. But in a hypoxic state, when you activate HIF1, you get more VEGF, you promote suppressive TAMs, you activate Tregs. Again, and you'll hear me say this a million times, it's just the wound healing process gone wrong. And you also activate disordered lymphatic vessels like VEGF-C and PDGF-B. And these bind to VEGFRs and to NRPs, which can then promote suppressive immune responses. So then you get this accumulation of these tumor-associated macrophages and cancer-associated fibroblasts that occurs primarily in the hypoxic core of the tumor and on the leading edge of the tumor, leaving the middle of the tumor, the epithelial phenotypic cells, lots of room to divide and not be attacked by the immune system. And this is going to continue to rise in importance because mature tertiary lymphoid structures are one of the clearest markers of anti-PD-1 response that I have ever seen. So this is a beautiful, mature tertiary lymphoid structure. You can see that it looks like a lymph node, but it's inside a tumor. And if you have mature tertiary lymphoid structures and you have non-small cell lung cancer, your chance of survival is much better than if you have an immature or no TLS. And then they show a patient here that has what's called an immature TLS, and you can see that your B cells are not arranged in that same for CD20. They're not arranged in the fashion that they are here. You know, your CD8 T cells are all over the place. They're not interacting with each other. And over here, they're neatly organized. So this is a patient that's in this group with mature TLS, and this is a patient who has immature TLS in their tumor. And that's what happens when they get immunotherapy. It's like obvious disease progression. So if you can't form a tertiary lymphoid structure inside a tumor, you can't get clonal expansion where you recognize some of these subclonal antigens that we mentioned before, and ergo you can't get tumor clearance. So these are probably necessary, though possibly not sufficient, and a very, very interesting area of research. We'll probably have to add a B cell section and like the formation of TLS later on and all that kind of stuff.
So does that structure to exist inside the tumor, or can that exist in a lymph node?
No, it's inside the tumor.
Inside.
Yeah, that's what defines as a tertiary lymphoid structure rather than a primary or secondary lymphoid structure. And you do see them form in like rejected organs as well, but they're less well defined in that setting. So Tulia Bruno is an awesome a biomarker person who has done a lot of this. She's done a lot of this work at Pitt too, so she's in the same place Dario Vignali is, and great reading on TLS. But she's got a full biomarker guide of like what defines TLS and has worked on it with SITC. So, other thoughts or questions on TLS? Yes, yeah, great question. So Tregs, we've talked about Tregs a little bit. They require TGF-beta. This can come from a lot of different sources. Again, they have high levels of CTLA-4 and VEGFs. They steal IL-2. They suppress CD8 T cells directly. They're stimulated by IL-2, which limits this drug's efficacy and has made it such a problem over the years. And they also express similar exhaustion states to what Dan was mentioning before. So this is a really, I think, difficult concept for I even had trouble wrapping my head around this, to be honest with you, because it's like, okay, well, I'm going to block PD-1, but these Tregs express PD-1 too. It's like, should I be doing that? Like, so it's a very weird balance in the immune system, and so that's why I think like some of these FC modified second generation CTLA-4 inhibitors that allow for the clearance of these T regulatory cells are so important, because they're just like clearing the field of TCR dominance by Tregs in a tumor and allowing a normal process of tumor elimination to occur. So Tregs, really, really important. Treg targeting is something that's going to continue to be brought up. A tumor-tolerant stroma has been shown to generate antigen-specific Tregs, and this is like one of my favorite articles in like the last 10 years, because they show that a cancer-associated fibroblast-rich tumor microenvironment is just also chock full of Tregs. So they're like, well, I wonder if this could mean that the Tregs are actually TCR specific, and they're specifically instructing the immune system that this is self. And indeed they are. So if you don't use OVA, which is a synthetic peptide on an ovalbumin expressing tumor, and then you associate it with fibroblasts, the Tregs don't associate with the fibroblasts unless the OVA is there. So this is a TCR specific interaction. So you actually have pro-tumor TCR specific T regulatory cells. And where are they coming from? Cancer-associated fibroblasts that are actually presenting the antigen to them. So this is very paradigm shifting, I think, in this setting, and has a lot of implications for both targeting Tregs and cancer-associated fibroblasts. And again, I think this is a great paper, and it's a really interesting one from a spatial standpoint. I'm going to move right into monocytes, macrophages. The monocytes are in the blood, and they differentiate into macrophages in tissue. They make up the majority of the immune cells in the microenvironment, but we have no FDA-targeted therapeutics towards them, which is remarkable. And you can see that we have way reduced the complexity going into macrophage biology, because when you open up the macrophage compartment with single-cell RNA-seq, there's a minimum of at least 6, and I've seen upwards of like 12 different tumor-associated macrophage subsets that are across different tumor types, and many of them are conserved, and many of them only occur in cancer settings, which we'll talk about. So you can't just wipe out macrophages, because there's pro-tumor and then there's anti-tumor macrophages. So this is a real problem, because CSF1 targeting, CCR2 targeting have all failed. BMS has a graveyard of failed drugs in this area. And if you have these high IL-1B TAMs as opposed to just TAMs in general, you have much worse survival in pancreatic cancer patients. So the subtype of macrophage is extremely important. And we used to reduce these cells down to M1 and M2, which every time I see that in a publication after like 2020, I'm like, how do you excuse this in the era of single cell RNA-seq? Because like that's my single cell data up there in the top right. And I was just like, oh, okay, these are just the myeloid cells. Cool. There's a lot of them. And you can see those little pointillism-esque dots are the way that genes associate with one another. And each one is an individual cell. And what's interesting about this is we did not detect SPP1 positive or CXCL9 positive macrophages and associate them with immune checkpoint inhibitor resistance until this science paper from, I believe, the Schrum lab that was published a couple of years ago, using single cell technologies, which were able to say, hey, if the TAMs, the tumor-associated macrophages specifically, express SPP1, you're much less likely to respond to immunotherapy. And if you do a ratio of SPP1 to CXCL9 in macrophages, again, specifically, you can see that line of predicting checkpoint inhibitor response. If that's clinical outcomes, worse, better, more CXCL9 macrophages, better. And we didn't detect this for a long time because they're very hard proteins to detect, and they needed to be looked at specifically in this macrophage population. Again, you gain these SPP1 positive macrophages or osteopontin positive macrophages in a tumor. They don't exist very much in normal colon, as you can see there. Same thing with these NLRP3 TAMs, and you lose a lot of these IL-4I1 TAMs. The IL-4I1 is a tryptophan degrading enzyme, but in this setting, it's likely just a hallmark of phagocytosis. And you can see as you go further out from the tumor, your macrophage phenotype changes. And I don't mean to belie the complexity of that, but that then leads to saying you can look at IL-4I1 TAMs versus SPP1 TAMs, and you can predict clinical outcome in a way that's much more sophisticated than looking at one gene or a gene signature, something like that. So the diversity of macrophages leads to a diversity of immune suppressive mechanisms. So I'm throwing this up there kind of very flippantly just to go through it, but essentially you see macrophages that are existing in tissue repair that are promoting Th1, they're promoting CTL infiltration. This is occurring in normoxia. There's not a lot of extracellular matrix there, as opposed to ones that are acting in immune suppression and going towards fibrosis and tissue remodeling. And these TAMs produce IL-10, they produce TGF-beta, they promote those cancer-associated fibroblasts that then stimulate T regulatory cells. So they're really all part of this axis. The other thing that macrophages do is they're garbage at presenting antigens. Once again, they're just not as good as dendritic cells. I mean, what are you going to do? So the T cells have this short-lived interaction with dendritic cells. They go in, they get their message, they get their co-stimulation, they go do their business. TAMs hold them. And I thought this is like one of the coolest figures to me, because you can see that the tumor-associated macrophages have a less intense but a longer interaction with CD8 T cells, preventing their proliferation. So they're just arresting CD8 T cells. And so that's why when you have a patient that has a liver metastasis, and that liver metastasis expresses or exhibits lots of these SPP1 macrophages, what's happening? Like all these CD8 T cells are attached to it. They can't touch the poor lonely C over here. And all of the patient's blood volume is going through their liver. So that means their entire liver or their entire blood volume is being exposed to tumor tolerant factors between SPP1 macrophages, cancer-associated fibroblasts, and Tregs. And that tolerizes the entire body to the tumor because, again, your entire blood volume is going through. So this is a very important interaction, and I thought that this paper was really telling about like what these cells are doing on multiple different levels and how they are sequestering them from DCs. And this is a physical interaction. This is not determinable without spatial technology, really. And so I did that a little bit. I think the only thing that I would add to this is that you see fewer T cells in the liver mets versus primary. Oh, Ryan ditched us, so I was going to tell him that metastases are genomically identical, but they're different in their transcriptional program and microenvironment. This is why you need to know this. And there's some other examples of like pretty microenvironment pictures and how you can separate patients accordingly. We know that brain and bone metastases are also immune related sites, or immune privileged sites that we have trouble getting immune responses. And the brain is a little bit more difficult here because, as Dan mentioned before, this was often thought to just be a barrier issue, but there's obviously a lot more going on. I would note that a lot of drugs do not cross the blood-brain barrier. Like one very unusual thing we've seen is that Galactobart, which has an MMP domain attached to it, doesn't cross the blood-brain barrier as well. And we've actually seen relapses in some patients where we don't see that with botensilimab. It's a bit anecdotal, but I think it's important to what you were saying before with antibody designs. Like, where do you want your antibody to go? Are you creating places where the tumor as a privileged site? Are you really giving systemic therapy? So that's, I guess, the takeaway here. There are these VISTA positive TAMs, which produce these poorly described factors like midkine that suppress CD8 T cells. So I think there's going to be some unique mechanisms of immune suppression that we're able to appreciate in the brain. And this is something I think to pay attention to, especially if it's a research interest of yours, because there's a lot to learn there. The bone's crazy. I mean, the tumor just has everything, right? It's got all the myeloid cells at once there. So it can just activate granulopoiesis and send them everywhere. We know these patients don't respond well to immunotherapy. And there's just a paper, like as Dan and I laughed about this a few times, like as we're reviewing the slides for this, we're like, oh man, another paper got published. Okay, we got to add that one too. So there's SPP1 positive osteoclasts, and they probably sequester CD8 T cells in the bone marrow and prevent them from going out and doing their business as they do in the liver. It also promotes Th17 cells, which we talked about before. So you can see how when you learn some of the immunology earlier in this lecture and then you move it into different settings, it really hopefully solidifies that concept and how these different interactions occur in different systems. And this is really in bone and a great example of like a feed forward mechanism, because the tumors are facilitating IL-17 and TGF-beta, and then they're promoting these SPP1 osteoclasts through mechanisms we don't quite fully understand. And then those are producing more TGF-beta, and the entire cycle is feed forward. So unless we have a way to break that cycle, we're probably not going to overcome immunotherapy resistance in most of these patients. So I really think that these spatial relationships, if you think of like rearranging them, like the bispecific, being like the arranged marriage between the T cell and the tumor, and then like how does that look? What is it actually doing just to get them close to each other? Actually leads to killing. It tells you why antibody design is so important and why cell-specific type and interaction are so important. If you're very, very interested in biomarkers, SITC, the Society for Immunotherapy and Cancer, has a biomarker guidelines that we put in there that Tulia and some others have written, and it's really good. It's nice if you just need a reference to understand why somebody's doing the biomarker they're doing in a clinical study, or how you could possibly develop a biomarker out of the lab. So I think it has a lot of application to a lot of different people.
Yeah, I will too. Okay. All right. right? So this is a little bit of changing gears, but there's going to be some common themes, and we'll pull in some things that we talked about before. This is really going to take you back to college biology, because a lot of what is in this section is related to molecular metabolism and how tumor cells are able to leverage some of the molecular mechanisms that are normally used by immune cells and kind of starve immune cells, for lack of a better way of putting that. So number one to talk about is glucose and secondarily glutamine. What we know about cancer cells is they preferentially undergo glycolysis regardless of the oxygen conditions surrounding the cell. So any normal healthy cell, whether it's immune cell or any other cell in your body, is going to undergo oxidative phosphorylation. You get a much bigger bang for your buck in terms of ATP production with oxidative phosphorylation. But glycolysis is preferred by tumor cells, and this is what's been known as the Warburg effect for quite a long time. There's probably a couple reasons that tumor cells choose to do this. Number one, it's a massive starvation mechanism of other immune cells or immune cells in general from glucose. Number two, a byproduct that is produced is lactate. We'll talk about the suppressive effects of lactate here in just a second. But also, they have other mechanisms of feeding some of these further metabolic processes like the TCA cycle. So typically in a healthy cell, it would undergo glycolysis, then the TCA cycle, then oxidative phosphorylation. These all feed into one another. Basically what tumor cells are able to do is they do glycolysis on its own. They produce lactate, and lactate is lactate. It's done. They're able to feed the TCA cycle with glutamine, and they can keep the TCA cycle going with glutamine without needing any other metabolites. And the reason that they're able to do this is they substantially upregulate a receptor or a transporter really called GLUT1. And GLUT1 pulls in glutamine into the cell in very high rates. This is not normal. And so this is how they get around some of the need for moving directly into the TCA cycle. It allows them to produce lactate. Some of the therapeutic implications, that have been looked at without great success, can you augment the uptake of glutamine? Or theoretically glucose, but I think there's much greater side effects of doing that. But could you affect the GLUT1 receptor? It's been studied. The effects have been variable, as you can imagine. It's really hard to target one specific building block or metabolite, right? The other thing that has been looked at with more success is modulating the mTOR pathway. So this is an important pathway in metabolism in these cells. And it's using something like sirolimus has been shown to actually have beneficial effects on CAR-T because of the effects on metabolism, especially of the innate T cells, right? It's not necessarily even a cancer process in that case. So I've talked a lot about lactate. Lactate's important. Number one reason why it's important is it acidifies the tumor microenvironment. So we know that in tumor microenvironments that have high degrees of lactate, you see a pH of anywhere between 6.3 and 6.9. So substantially below what the normal physiological pH is. And the other thing that we know about that downstream is there are direct effects on CD8 positive T cells as a result of that acidification. The Delgoff lab at Pitt has done a ton in terms of lactate production mechanisms and the downstream effects of it. But one of the things that has been talked about quite a bit is this process of the lactate shuttle. So I mentioned that glycolysis occurs, lactate is spit out. That lactate is actually shipped out of the cell and it's moved primarily to T regulatory cells. And so these tumor cells are essentially feeding their immunosuppressive counterparts, allowing them to exist in great happiness. And what we know about lactate-avid T regulatory cells is they are much more effective T regulatory cells than glycolytic T regulatory cells. So there are two subtypes. What you see around tumors is there's a much higher degree of your lactate-avid T regulatory cells than your glycolytic T regulatory cells. When I talk about differential effects on CD8 positive cells, again, you see decreased efficacy of your CD8 positive T cells at acidic pHs. So there's been theorized, could you buffer the tumor microenvironment by doing something like adding sodium lactate instead of lactate itself and bringing the pH up? And what they've actually shown in the lab is increased efficacy, or really you're pulling T cells out of energic states and allowing them to act by buffering the tumor microenvironment, which is quite unique. But again, you run into the logistical issues in vivo, right? The other thought that's a major one in this space is, can you block some of the enzymes with the production of lactate, something like LDH, which oximate is important for. has been met with varying degrees of acceptance. So here again, reinforcing all the differential effects of lactate on the tumor microenvironment as well as immune cells. Generally bad. I just wanted to introduce this concept. We'll talk about it a little bit more because it's more relevant for fatty acid oxidation and lipid production. But you see there's so much interplay between the TCA cycle and fatty acid oxidation over here. And some of the mechanisms that cancer is able to use to get around not needing to go through all of these processes is sharing of metabolites across the board, really. And lactate is the biggest one. Talk about that here in just a second. But this is just reinforcing the fact of what lactate can do in the tumor microenvironment with a specific recently published paper looking at how tumor-derived lactate affects this SREBP2. And what it does is it affects SREBP2 by activating it in the tumor microenvironment. And what we know that does downstream is it affects MREG DCs. Which Nick has already talked about. These MREG DCs downstream, of course, we know suppress CD8 function, increase regulatory T cell function. So immunosuppressive across the board. Okay, fatty acid synthesis. This is important both for immune cells and tumor cells. It's important for immune cells because when immune cells are starved of glucose, they will undergo fatty acid oxidation. That fatty acid oxidation leads to a variety of different things, but obviously, some of that is production of fatty acids, and you can see all of the differential effects that fatty acids have on the immune system over here. So in your anti-tumor immune cells on the left, all of the different blocking effects of things like cholesterol, for example. And then you see the pro-tumor effects on the other side. And I think that's a little bit better illustrated on this slide in terms of what that looks like when it comes to lipid droplets. So the production of lipids from fatty acid oxidation has a variety of downstream effects. Number one is the fact that we know that the phospholipid bilayer is buffed up in these tumor cells such that it makes it, in certain circumstances, more difficult for chemo to work because chemo is so reliant upon getting into the cell, affecting different things within the cell, whatever that machinery may be. This lipid buffering, for lack of a better word, is very important in that process. The other thing to think about is that lipids in immune cells equally affect the efficacy of them by preventing things like appropriate antigen presentation in APCs. I have some red boxes on the slide here. These are possible targetable areas of the fatty acid pathway. I think one of the ones to point out that's really interesting is HMG-CoA reductase, which everybody in this room probably knows is being associated with statins, right? And so there's some data, some theoretical benefit as well of using statins to potentially downstream affect the prevention of cholesterol production, which then downstream prevents lipid droplets from forming and theoretical benefit with better efficacy of chemotherapy. The other one to talk about is CD36 at the top up there. This is basically a transporter that pulls exogenous fatty acids into the tumor cell. Or in the case of some of the studies that I'll talk about, T cells, what we know about this CD36 is it can pull in massive amounts of fatty acids, which, you want some because that cell has to undergo fatty acid oxidation, but you don't want a ton because we talked about the negative effects of it. So there's been some studies to look at blocking of CD36 in combination with PD-1 with actually decent benefit in doing so.
How are the lipid droplets preventing antigen presentation?
That's still not totally known, but it's well mostly thought of as that when you have antigen cross presentation, it requires lipid rafts to shuttle the MHC complex as it's after it's formed in the endosome to shuttle it to the cell surface. So it's possible that just like the sheer fact of having too many lipids just makes for fewer MHC complexes that are antigen loaded getting to the surface. Is really hard to know. We know that that SREB program that Michael has described really leads to like far fewer antigen or far less CD8 cell proliferation and far more T proliferation. So it's clearly there's more to it than just that. Right? Very much.
So we talked a little bit about ATP and adenosine earlier, or well, ATP specifically. It's obviously an important molecule for energy, but again, when we talk about some of the mechanisms that tumor cells are able to hijack in comparison to normal immune system, this is another one in that they create large amounts of adenosine by breaking down ATP. And adenosine has a variety of different effects, but we know that it inhibits healthy CD8s and CD4 T cell function, and it causes Treg expansion. And so there's been a few studies that have looked at the possibility of using a CD73 inhibitor, which is an important portion of the AMP to adenosine conversion process, and whether or not that would actually affect the immunosuppressive effects, right, that adenosine could theoretically cause by preventing its breakdown. But A2AR, which is an adenosine receptor, has also been studied as well, and again, some potential benefit in this setting. Okay, IDO and interferon gamma, and we've talked a lot about interferon gamma, and it's a nasty player in this setting because of the fact that it induces IDO1 expression. And if you remember about interferon gamma, it's actually released by your your T cells. And the problem with that is by inducing interferon gamma, you're causing, on the right-hand side over there, you can see the breakdown of tryptophan into kynurenine. And tryptophan, of course, is an important portion of the process for T cell activation. And so by starving the T cells of tryptophan, cause major issues. And so IDO1 as an enzyme is important in this process. The other thing to mention, though, is that there's an enzyme on the other side of this, a TDO, that can actually basically play a duplicative role to IDO1, which is important to keep in mind. We know that kynurenine is immunosuppressive, binds to this aryl hydrocarbon receptor with IL-4I1, and downstream leads to a lot of different issues. So theoretically, if you could block IDO1, the question is, could you actually have therapeutic benefit from that? That has been tried with epacadostat. I won't say anything more about that right now. Okay, lastly, to wrap things up on a specific level, arginine metabolism. We talked a little bit about M1s and M2s already. We know that M1s are pro-inflammatory, M2s are immunosuppressive. That's oversimplification, but we know that your immunosuppressive macrophages have a tendency to overexpress ARG1 as an enzyme to break down arginine, whereas your M1 and your general good macrophages overexpress or express this nitrous oxide breakdown enzyme called INOS. And you see the differential effects of doing so. You know that arginine either way is an important, well, it's a semi-important amino acid, but you see the effects of either going through the INOS pathway versus the ARG1 pathway and the starving on the M2 side. Okay, so pulling all this together, you can see on the left-hand side, you know, normal cell, very simple process, right? We talked about glycolysis, TCA, oxidative phosphorylation, energy is produced. That's all that needs to happen. You can see all the different ways that the cancer cell is able to hijack this, though, right? So the GLUT receptors on the bottom, the shuttling of lactate over here on the right-hand side, the variable moving of lactate back to pyruvate into the TCA cycle in certain circumstances. There's a lot of these mechanisms that get very messed up in tumor cells. And so, you know, a lot of potential druggable targets, though, too. The problem is by doing so, you could theoretically affect healthy cells as well in certain circumstances. All right.
All right. So we've got a good amount of time to go through these last ones and then probably do, like, I would say, a 20-minute rundown through everybody's articles and activities that we sent out, which will be like very brief and informal. If you want to draw stuff, you can draw stuff, whatever you want to do. So the last two sections are on the extracellular matrix and tumor intrinsic factors of immunotherapy resistance. And I think that the extracellular matrix is a pretty difficult topic to approach because it's so poorly studied. So I put this picture of like a decrepit house in like a post-apocalyptic setting, and just like imagine if someone told you, "Hey, can you go get a jar of mayonnaise?" You'd be like, "What? How am I gonna? I'm not gonna do that." And the mayonnaise can't be good. Like, I mean, that's a terrible idea. So that's what it's like getting through the extracellular matrix to actually like kill a tumor cell. It has a large amount of deposition, decreased porosity, increased stiffness that prevents immune cells physically from getting across. And these are formed by these lysine residues they're also not only just a physical barrier for migration, but they also alter immune cell function and cancer cell function. So again, I'm emphasizing cancer as a chronic non-healing wound because there's a lot of fibroblasts and extracellular matrix deposition there that's not going anywhere. And interestingly enough, these fibroblasts and extracellular matrix compositions can restrain or promote tumor growth or metastatic progression. They can prevent therapeutics from getting in there, and they can certainly interfere with immune function. So all of these cells can modulate the extracellular matrix. And we talk about fibroblasts a lot, but it's like, what even is a fibroblast, man? They're these mesenchymal mobile cells that are important in tissue repair, that are capable of phenotypic differentiation, antigen presentation, as we talked about before. So tumors mobilize these cells, they imitate them, they copy their processes, and they deposit hyaluronin, fibronectin, collagen, and this dense extracellular matrix, which also sequesters TGF-beta, which, as we've kind of talked about a little bit, is a broadly immunosuppressive signaling molecule. And the extracellular matrix, which was long thought to just be, it's just goo around the tumor, like you just need to, don't worry about that. It actually interacts with a lot of like integrins signaling through FAC pathways, and can interact with CD44, which is a stemness pathway. So this is probably more important than we necessarily appreciate now in like metastatic site outgrowth. I think one of the highest possibilities for targets in the near future is silation and glycosylation. These are post-translational modifications that we don't spend a lot of time on because it's not RNA, DNA, it's not protein, it's carbohydrates. And they stick to the outside of receptors in the extracellular space, and they prevent their binding. So it's like, how are you going to target PD-L1 if it's just like chock full of glycosaminoglycans? So you're not, and this is true of a lot of other receptors. This can happen with antigens as well. So this is a way that tumors can evade, even if they have a clonal antigen, that they are actually able to cover it up, essentially. GD2 is probably the best example. There are CAR T cells that have been developed against GD2. So these are independent targets in and of themselves as well. And they're broken down into kind of like all of these different categories. And you can see, again, this is just really to demonstrate the complexity, not to get you to memorize exactly what selectins and Siglecs and other things are. But in general, I think the best way to think about this is that you have these carbohydrate-laden extracellular matrix, proteins that interact with other cells, and it is both indicative of like the immune microenvironment and the tumor itself. It can lead to direct migration, tissue homeostasis. This is an area where there is substantial differences between mouse and human. There's very little overlap. So these are also very hard to study for this reason. So it just adds another layer of complexity to it. CD22 is an example of a Siglec, I think, that's being targeted, and there are some others that are out there too, like MGL is GD2, et cetera. But all of these, again, are protein carbohydrate complexes, and they're a mechanism of immunotherapy resistance. And again, there are multiple different ways that these can be targeted. Targeting the Siglecs themselves, combining drugs that inhibit cancer glycosylation, and modulation of upstream signaling. And they can also be used for biomarkers. I think this is an overlooked biomarker space. And that is probably one of the most interesting things to me, is that like we have so much trouble identifying who is going to respond and not respond to therapy. And it's like, well, here's a whole nother layer we haven't been looking at, So silation in particular, like studying these extracellular proteins with silyl groups, studying them with carbohydrates, you can split patients up into different subtypes. And if they have a low level of silation, whether they have kidney cancer or lung cancer, it doesn't matter. They do much better than if they have a high level of silation. And the high level of silation is also correlated with, stop me if you've heard this before, cancer-associated fibroblasts, macrophages, and T regulatory cells. There they are. And so you have this strong correlation. It's like, how can you possibly overcome this? And in this study, they inhibited silation with a sialidase enzyme. And I thought this was like, a really elegant way of doing it because, well, in this case, they're improving anti-PD-1 outcomes. and anti-CTLA-4 in their mouse models. And we could talk about their mouse models another time. But the point is that they're seeing an interferon gamma specific response because they're actually able to get T cell recognition of the tumor just by removing carbohydrates from the outside of the tumor. So how can we change existing therapies to overcome silation? And this is where man, when she presented this, as Carolyn Bertozzi presented this, I was like so excited. She's like, it's like a lawnmower. And then she shows like a GIF of it, like shaving off all of the sugars on the top of the cell, and then trastuzumab can then reach its receptor. So you see, if you have an active sialidase, how much more trastuzumab is binding to the cell than if there isn't one. And so it was really hard to ignore that effect. Increasing the availability of these surface proteins may inhibit complement activation and ADCP, and this reprograms those tumor macrophages. So you can take existing off-the-shelf therapies and potentially make them much more effective just by slapping a sialidase in the back. So lawnmower map, all about it. Other ECM components that are orchestrated by tumors, platelets and neutrophils are known to escort tumor cells, and this may be prompted by stress states or therapeutic pressure. They assist in the deposition of ECM along with fibroblasts, and they contribute to fibrosis and thrombosis. So I think if you're on the heme side of things, this is something to really think about because there's not a lot of data out there for this. So there's a big opportunity to learn more. There's other mechanisms of metastasis that occur and that could be influenced by ECM that I've listed there. So they all require a soil for the seed. The tumor has to have a place to go that it can actually grow. So that means you need to have a tumor microenvironment in ECM that is capable of doing that. Understanding how the tumor does that is sort of the next step here, because otherwise we're not going to figure out how to overcome this process long term. But I think the sialidase modification, in all seriousness, is a really great, readily available way to potentially improve therapeutics. I do hate me. There we go. Oh, let's talk about nets. That's right. So these are just, I don't know, I just find these to be gross, to be honest. Like neutrophils just extrude their DNA. It's like just very, ew, how rude. But if you have lots of nets in an area, then you get a higher outgrowth of tumors. So in patients that are smoking cigarettes and they have a dormant tumor, a neutrophil extracellular trap stimulated by the inflammatory effects of the tobacco smoke actually is what leads to the tumor outgrowth, not the mutagenesis itself. So I thought that was like very fascinating. And the same thing happens with chemotherapy through IL-1B, which leads to neutrophil recruitment, and then you get release of active TGF-beta, and then you get this extracellular matrix deposition. So nets are easy to detect by biomarkers. You can stain for them. These are not difficult from that perspective. It's just a matter of better understanding how we can target them. I'm going to kind of close out on a little bit more of cancer-associated fibroblasts because they're just so incredibly important, and we don't have targeting for them yet. And I want to convey like the heterogeneity of them and how they interact with all of these different immune cells and can contribute not only to that, but tumor vascularization, innervation, barriers, selection, essentially creating an immune privileged site. So I think that these probably have a lot to do with the formation or lack of formation of tertiary lymphoid structures as well that we don't quite understand. But if you have these cancer-associated fibroblasts that express a protein called FAP, which is fibroblast activating protein, and you have more of those SPP1 macrophages close to those fibroblasts, then you have a much worse prognosis in colorectal cancer than if you don't. I mean, that's a very, very stark difference there, just looking at the TCGA, which is admittedly not the cleanest data set. So more FAP high fibroblasts, more SPP1 macrophages, they're working together with each other, and they sustain a niche that sequesters CD8 T cells from CDC1s, as we talked about before. So while they can also have their own antigen presenting functions, they can promote the terminal exhaustion of CD8 T cells, and this occurs through dysfunctional dendritic cells. And thus they are orchestrators of the immune desert. And one of the mechanisms that this can occur is in response to therapy and also in response to toxins, but in response to aging. So there's a different mechanism that has been described in melanoma of metastasis emergence from dormancy in young versus old patients, and it is completely dictated by the state of the fibroblasts in the tumor cell. So I think there's a lot more to learn here. This is meant to just kind of like dip your toe in the water of cancer-associated fibroblasts. I think I'll skip this, but this is one way that CAFs can directly suppress NK cells. So this is more just to express the fact that we have lots of ways that we could potentially target them, but they haven't done well at all. All of these trials are failures that I listed up there, and they're probably due to redundant mechanisms or other things that we haven't talked about yet or will talk about. One of the most promising targets here, other than FAP, I think, is CXCR4, which is on tumor cells and binds CXCL2 from fibroblasts. And this is thought to be like a positive feedback loop that leads to tumor cells promoting like Wnt and NF-kappa B signal. There's just so much more to learn here. And I think that it's one of those things that if you're interested in cancer-associated fibroblasts and you want to study them, or you come across a drug that's targeting them, I would really spend time on learning a bit more. Okay, I'll pause, and then we'll go into tumor intrinsic mechanisms and finish it out. All right. This is my hedgehog. He suppresses the immune system. I haven't named him yet. But I think it's really, I put this up there because an AI made it for me. And BLE2, which is part of the hedgehog pathway, suppresses anti-tumor immunity, and it is a tumor intrinsic mechanism. So while we've covered like all of these different mechanisms of tumor intrinsic immune suppression, how does a chronically inflamed tumor survive? What are the impacts of tumor mutations? How do tumors evade the immune system on an intrinsic level by like recapitulating fetal programs and promoting EMT, stemness, and other immune privilege factors. So mutations orient microenvironments, and again, like this is a nice review that we put up there, but it's like as soon as you open this review on mutations orienting microenvironments, it's outdated because something else has come up. So I'm going to hit a few examples here, but not a lot, because there are many, many more that we could go into, and this is a lecture in and of itself. So KRAS itself, just activating the MAP kinase pathway, and it's hard to know if this is KRAS G12D specific or if there are other MAP kinase mutations that would affect the microenvironment differently. That's a very interesting question. But a KRAS mutation will facilitate the production of TNF alpha and IL-1, and this recruits these STAT3, IL-33 promoting fibroblasts that then recruit those Tregs and likely lead to a similar mechanism of immune suppression like we described before with SPP1 macrophages and TCR-specific Tregs. KRAS has also been shown to recruit PMN MDSCs. So I think this has a lot of implications, and it's easy to look at it and say, oh, of course, you mutate a gene that's going to lead to all these immunosuppressive, downstream effects. But it's like these are something we have a readily available biomarker for and potentially downstream targetable pathways. So I loved the way Ben Stenger put this at AACRI this year. He's like, some oncologists don't even know they're practicing immunotherapy, but you are, buddy. Sorry to tell you, you are. There are several papers that support immune requirements for full durable MAP kinase inhibition. You can get enhanced MAP kinase inhibition with immunotherapy. You don't necessarily see the same thing in mice as you do in humans there. So I do think that this needs to be carefully trialed in humans in sequenced settings. And one of the reasons that is, and this is one of those concepts, if you take very little away from this lecture, I really would make sure this one sits in. If you give a MAP kinase inhibitor, then you enter a brief period of immune activation. And as you develop resistance to that MAP kinase inhibitor, it has cross-resistance to melanoma. And this has been shown, or cross-resistance to immunotherapy in melanoma. And this has been shown clinically in the DREAMseq trial, which is an elegant, well-executed, smart trial. This is the kind of stuff we should be doing. So nivolumab, ipilimumab first, followed by dab-tram versus dab-tram, followed by nivo-ipi. So people are afraid to use immunotherapy first because they're afraid that the response isn't going to be fast enough. This takes away a little bit of that anxiety, I think, because you see the 2-year overall survival has a 20% difference. You give the immunotherapy first, you get your durable response, and then you use your MAP kinase inhibition as a backup plan. Maybe in the future, we'll be able to drug MAP kinase for a short period of time, come in with anti-PD-1, but that's the kind of stuff we need to figure out. And DreamSeq made an immediate impact and changed the way particularly community oncologists practice. So great trial, really emphasize that one. Other things that are really important when it comes to tumor intrinsic immunity, we know that P53 mutations and P53 loss both promote the production of Wnt ligands, which lead to suppressive macrophages. We'll talk a little bit more about what Wnt ligands do later on. P10 has been associated with production of VEGF and clearly is associated with a lower response to anti-PD-1 therapy, but it doesn't really seem like we have a great handle on that. I think there might be other mechanisms of resistance because VEGF plus anti-PD-1 hasn't really panned out in these settings. So there are probably, and this is true of every pathway that we've talked about so far, P53, MAP kinase, et cetera, there are probably multiple mechanisms of immune suppression. It's probably not just VEGF, it's probably other ones too. So targeting one is kind of like playing whack-a-mole in this setting, which I think is why the idea of giving these targeted inhibitors in like a window setting is really attractive because it's like maybe you're able to drop all these immune suppressive mechanisms briefly, have a window of anti-tumor immunity. And as you expose tumors to interferon gamma, you want there to be some interferon gamma, right? Because otherwise it's this immune desert. And so when you get these immune responses, when you stimulate the anti-tumor immunity, what does the tumor do in response to that? So if you have no recognition, you have, again, this immune desert. Ideally, you have this controlled functional interferon gamma expression. The immune system likes to take care of things, it likes to complete stress cycles, if you will. It wants the tumor to go away, it wants to produce less interferon gamma, and then it wants to go about its business. And if you have lots and lots and lots of interferon around, you start to get NF-kappa B signaling. You start to get chronic sting activation. This enables a switch, an epigenetic rewiring that leads to a more immune suppressive microenvironment. So this is yet another mechanism of tumor intrinsic immune suppression, and this is one that is not due to a mutation, but is tumor intrinsic. And chromosomal instability can also induce this CGAS-mediated sting. So this is kind of like with Afrin and tachyphylaxis, where if you just like keep giving Afrin again, like it won't work anymore, So who else is a sinus sufferer? Show me. Tachyphylaxis to interferon is the same thing, and this occurs to be a ER stress. So if you have chronic sting activation and a chromosomally unstable tumor, you don't want more sting around. So why have sting agonists failed? Samuel Bakun's work really demonstrates that really nicely, and that you get promotion of metastasis in response to intrinsic chronic interferon gamma signaling and this massive upregulation in EMT or epithelial to mesenchymal transition. Cells also remodel their epigenetic expression by TCF-BRD4 or EZH2. This is a very common way that they'll decrease antigen expression. I won't go into this too much, but I think that there are a lot of cross-resistance mechanisms to therapy that are occurring here, and that these epigenetic modulators, and there's some very good papers on EZH2 targeting. I just don't really know how it's panned out in the clinic yet, which is kind of a problem. So I would really like to see this develop more because it's known to increase the antigenicity of tumors and potentially affect other immunomodulatory pathways. Much more to learn here, both on a tumor intrinsic level and I would argue on like an immune level too. We know these tumors undergo something called oncofetal reprogramming, which is a term that I really like because I think it adequately describes what they're doing. Tumors have the entire genetic playbook at their disposal, right? They can do anything they want. They can access any gene under the sun. So that's what they do. So if you mutate beta-catenin, it suppresses CCL4, and then you get DC recruitment, or you lose your DC recruitment. But also, what we've shown in the lab is that transcriptional regulation, independent of mutations, of Wnt ligands, either through NLRP3 activation or hedgehog crosstalk, and others have shown with P53 mutation, you get an upregulation of IDO in a sustained manner that is more so than interferon gamma. You get increased kynurenine production and therefore T regulatory cells. And Wnt ligands also signal in a tumor intrinsic autocrine manner through the YAP pathway that produces CXCL2 and CXCL5, drawing in more PMN MDSCs. So you can see how these Wnt ligands have multipotent immune suppressive effects, and they're not regulated necessarily by a mutation, but can be regulated by NLRP3 activation, which is an inflammatory mediator, hedgehog crosstalk, which has multiple non-canonical pathways like TGF-beta and hypoxia, and P53 mutations itself. So Wnt ligands really central to a lot of immune suppressive processes and something we've spent a lot of time studying. I am not going to spend time on Notch because Notch is really confusing. I will say that there's a lot of stuff downstream of Notch, and probably one of the best papers, and I find this paper to be, very intricate. So it's one to read if you're interested in this pathway. Notch ligands are really important in like T cell development, so it's hard to target them. They're a difficult drug target, I think. But it's been shown that if you delete JAGD2, you restore Notch ligand upregulation, and that in turn leads to anti-tumor macrophages. So probably a lot more to learn here. I doubt that we'll go much longer without some sort of Notch immunotherapy combination. It's just probably going to be highly patient selective, and it's hard to know because the pathway is so poorly described. I've mentioned TGF-beta a million times. It sustains Tregs, it polarizes TAMs, it washes the dishes, it does everything. It activates fibroblasts. It serves as like a thermostat for EMT in normal settings. And this is the important concept I want to convey off this slide. It's supposed to slow cell growth down and force it into a more mesenchymal state. And the terminal state, if you just pour TGF-beta onto a pluripotent stem cells, it just becomes an adipocyte and stops doing anything. So what do cancers do? They make a balance of this partial EMT so they can really, manipulate it in their way and preserve their proliferation in these high TGF-beta environments. So that's, I think, really central conceptually to understanding, like, how does TGF-beta suppress immunity and promote tumor growth when it's supposed to slow cell growth down? So cell death is, again, a whole talk in and of itself. I put an immunogenic cell death definition on SITC guidelines. I think this is more of an academic reading on your own exercise. I won't go into the great details of tumor cell death. I think it's just important to realize that there is a balance here too, and that you can lead into suppression where your body is moving more towards like an a wound healing state. One of the ways that tumors do this is they co-opt NF-kappa B and TLR signaling. You can see the definitions of immunogenic cell death there. DAMPs and PAMPs and all of these other things that regulate TLRs, et cetera, they can also promote tumor intrinsic pathways. And one really nice example of this is the one I mentioned before with Wnt5a, wherein TLR4 promotes Wnt5a, this signals via YAP, and it promotes PMN MDSCs. But upstream, what's signaling via TLR4? Well, you have PDL1, which then downregulates STAT3, that downregulates PKR, and then it upregulates NLRP3, and then it produces HSP70, and that's what binds to TLR4. So it's actually the CD8 T cell killing and activation of the tumor cell that then leads to this intrinsic mechanism of immunotherapy resistance downstream of cell death. And I think that this is a complex, difficult mechanism, but this is the kind of ways we need to think of things like adaptive immunotherapy resistance and how tumors evade the immune system over time. Just harkening back to that evolutionary concept. We talked about FAST a little bit. I think this is really just to convey that tumors can kill T cells themselves just by expressing FAST ligand, and then they can also engage in, it should be called fratricide, shouldn't it? Maybe they shouldn't have gone with fratricide there. That's just easy. So they can also kill each other because these dying CD8 T cells will produce FAST ligand themselves. And I'm going to end on prostaglandin signaling, which I think is a prodigal example of cell death and stress that mediates a dual function. I mentioned before, you have these EP1, EP3, and EP2, EP4 immune stimulatory and immune suppressive receptors. So targeting COX-2 or some of the other mechanisms of prostaglandin signaling where you just eliminate PGE2 entirely, it's not surprising that you do not get dendritic cell recruitment in those settings, because that is the purpose of PGE2, is to bring dendritic cells and other antigen-presenting cells to the site. And after that, it stops being an immune stimulatory component. So constitutive expression of PGE2 in tumors is well described as an immune suppressive and pro-tumorigenic mechanism. Again, the net result is this direct suppression of NK cells, promotion of PMN MDSCs, which I showed before, and suppression of CXCL9 and DCs. So again, the net result of prostaglandin E2 in tumors is to lead to immune suppression. And it's promoted by multiple different pathways. So these are Western blots from our recent paper where it's showing if you activate GLE2 in the tumor, you get upregulation of COX-2, PGE2, and this whole downstream immune suppressive pathway. But if you knock out GLE2, it doesn't matter that you have both, in this cell line in particular, the PA3 kinase and the MAP kinase pathway active, you can't get COX-2 upregulation. So there's a lot of tumor-specific regulation of these immune modulators that we don't quite understand, and how could we better target them, possibly with drugs like TPST-1495 up there, that is a selective EP2, EP4 inhibitor that's worked pretty well in our hands. That's it. That's the talk. Good. We got like 10 minutes for questions plus doing these activities as much as you guys want to spare time for. I don't know how much you looked at the articles beforehand, or if you just want to talk through them now. But Bueller.
So that was phenomenal.
Cool.
Through to force.
Yeah.
Holy shit. A lot of stuff going on.
Yeah. Sure enough. Needs to be 2 talks, right? That's what I tell Andrea.
5 or 6.
Yeah.
But so one thing, and this is just kind of from the clinical translational perspective, is that given this extraordinary complexity, what do we ultimately do? Nivolumab, right?
Yeah.
And so what do you think are kind of the next big frontiers? And you discussed a lot of this throughout the presentation, but like, how do we take this knowledge and translate it into better outcomes? What are the big gaps where there's a huge need to take and reconcile all the complexity of the molecular aspects of this and make it into something that we put into a person that makes them better?
Phenomenal question. So this is, I'll answer this with an example. This is BBACO, our trial of botensilimab and balstilimab first line in MSS colorectal cancer. These patients haven't seen chemotherapy before. You're studying a naive tumor and exposing it to the immune system. We're getting pre and post-treatment biopsies and doing spatial analysis on them, so we better understand where the immune cells are and how they're associating with each other. And I think taking these broad viewpoints and recognizing how much we don't know, and like looking at the breadth of data that's out there, and then you have to bring that back to the lab and recycle that back into the clinic. And that's, I think, that kind of translational engine that you're referring to, it hasn't been revved up in immuno-oncology yet. Yeah. You know, it's really, I know it's easy to say, oh, we've got a CCI, of course, everything's fine, blah, blah, blah. But it's not. Like, there's still a lot of like Rego Ipi-Nivo out there, which is, I'm gonna get me started on that, man. Let's go. Regorafenib hits FLT3, so it blocks dendritic cells.
Right.
And you're gonna combine it with Ipi-Nivo. I described that to Hope Uranus, and she was like, "Oh, so it's like tying your shoes together and running a race." I'm like, "Bingo, Hope, you got it." So we can't keep doing stupid stuff. I'm sorry, but it's like that's the way to put it. You can't just keep throwing spaghetti at the wall and hoping that you're gonna get pasta. Like it's just not going to happen. Right. Like we really have to be rational in our combinations, and like the DreamSeq trial, you have to be willing to do these sequences and ask these questions. Like, man, the FDA gave us so much crap for doing BOT/BAL in patients with colon cancer. Like, you're depriving them of chemotherapy. I'm like, depriving. Yeah, that's not the word we want to use here. It's like, would you want oxaliplatin? No? Okay. So, I mean, we have to be bold in those kind of decisions, I think, and it's going to require a lot of challenging of norms. You know, STING agonists are another great example, like, of this. It's like BMS is still developing a STING agonist despite everything you've just seen. So, a lot to ask, and I think being smart and doing lab studies in parallel is what's going to really help.
And I think a very good point you raised here is that some of this, in principle, the most effective way to really understand this is to actually start with treatment naive patient population. And that's really hard to do when you have a good standard of care that already exists.
It's a hill I die on.
Yeah.
I'm serious, man. I don't know another way to put it. Like every time I see this patient scans, I'm like, that's why we did it. That's why we did it. Like, it was very hard. The FDA makes us scan at 6 weeks, which is absurd, but that's how you change it. And melanoma, I don't want to say it was easier, but it's like you had a very low bar. Like FOLFOX does save people's lives. I mean, like, let's be honest. Like we do cure people with FOLFOX that have like a oligometastatic disease. It's not like we should throw the baby out with the bathwater.
Right.
So it is a harder sell. But I think like as this goes on, as we kind of push like rational combinations and drive biomarker development and lab development at the same time as trials, this will start to fall into place. But there'll still be dumb. There'll still be so much dumb.
Oh, yeah.
Don't worry. Yeah.
Okay.
I was wondering, so I feel like when you did consultation in the fall, you had not a rant, but a good explanation.
It was a rant. Let's just be real. It was a rant. And it's the thing I did. It's fine. Yeah.
Of like why you think that I think PD-1 inhibition makes more sense than PD-L1.
Yeah.
Could you explain that again? Like
Sure. So this kind of attests to some of the things you were talking about before. So it's like you've got PD-L1 on your tumor, and you've also got APCs that express PD-L1 and PD-L2. In fact, PD-L2 is really highly expressed on those IMREG DC2s. Like when Michael would do flow for them, it was just like off the charts. He would always have to do an FMO for us to like really believe it. And then you've got your T cell, which has got PD-1, and it can go to PD-L1, it can go to PD-L2, it can go to PD-L1 of the tumor cell. It can also co-bind with FLAG3, as we mentioned before. So it's like, this is the central mediator, at least to me. And we'll have to take this out of the recording. But there's internal data at Roche that they have of a PD-1 inhibitor that outperforms atezolizumab. So there's no rationale for it. Like I use it when I have to, like when I'm forced into it, basically. I would say kind of a similar thing of some of the CTLA-4 regimens, like how they're playing out, So this is to your point too. It's like if we don't question these things, if we just roll it out to say it's just a slightly different drug target because we want a patent and a different indication, which I think is a question you always have to ask. Are they just fooling around with this to get a distinction because there's such a competitive space? But I mean, we all don't care, right? It could be called anti-PD-1 antibody. We'd be like, great, we don't need like Opdivo or something like that. So just keep that in mind of what their motivation is and what years. So great question. Other ones you guys want to talk about articles briefly?
I had a question about that experiment. So you had the mice where they knocked out for H2 to induce them as
Mm-hmm.
So you're saying because, just to make sure I understand, so it's because what you're getting is subclonal population.
Right.
As opposed to clones that the T cells are recognizing different
Yeah.
things. And then you said something about TCR sharing.
Mm-hmm.
Can you explain that and how that
Sure.
plays a role in that?
Sure. So as you go through class 1 and class 2 antigen processing, you need the CD4 T cells to have overlapping recognition of the same antigens as CD8 T cells, or you don't get that CD40 interaction from the Th1 cells, and therefore you don't get the second signal for co-stimulation.
Okay.
And so there's one possibility that Tregs sort of siphon off the TCR repertoire from these Th1 cells, since there are these tumor-specific Tregs. You'll notice that like a lot of things in immunology work in like, where is the pool of that molecule? Like IL-2, for example. Of all the IL-2s with the CD8 T cells groovy, it was with the Tregs problem, and that's why they will steal it from them. So similarly with this, and to make this even weirder, Tregs can undergo something called trogocytosis, which is where they steal a TCR or place a TCR onto another cell. It's so weird. So they can exchange receptors across the immune system, and DCs can do that too. So there's a lot to learn about PCR sharing, I think, but the main thing you should take away from this is like anything that has to do with like your Claudin 18.2 CAR T cell studies that you've so well explained in all your presentations, like they take CD3 cells, right? They don't take just CD8s, and that's because you need that TCR share. So they sacrifice the fact they're probably taking some Tregs to do that. But there's a lot more to kind of understand.
So that's referring to CD4 T cells and CD8 T cells have different TCRs, but they're just recognizing the same antigen.
Bingo.
But what you said with Tregs is that could actually be like literally the same TCR and a Treg, so they're clonally related.
Yeah.
To the T.
Yeah. So that's why if you do like single cell TCR-seq, you have to do it in pair and in concert with single cell RNA-seq. So you can differentiate where your TCRs are, and then you have to barcode them back. Cool. I'm gonna pull up the papers. We'll just kind of like run them down real quick. You guys have 5-10 minutes. We'll just talk about sliding scale. And if stuff comes up afterwards, please email us. It's not just the feedback. We also want to like foster ideas. Anybody want to speak up on their articles? Come on, somebody's brave.
I can explain mine.
Yes, do it. Yeah.
This is the hyaluronidase one.
Yeah.
So they combined hyaluronidase with gemcitabine for pancreatic cancer. The idea was that the ECM is like blocking tumor penetration with chemotherapy. And they selected for patients who had anything like 50% or higher hyaluronidase expression to select their population. But basically there was no difference between the group with the study versus with just chemo. I think in this trial, one of their explanations for that was by disrupting the ECM, they may, the idea is to increase accessibility of the chemo, but maybe they're also, the ECM may also play a role in sort of restricting tumor cell metastasis. And so it's really, I guess it's just that it's really not that straightforward that it's doing one thing, it's doing multiple things.
Perfect. Nailed it. No notes.
I was curious in that trial, I didn't see like, I don't know, maybe I just missed it because we were doing quickly. I didn't see any data that really showed that it was effective in terms of like CT scans that showed a decrease in the Schrum load. Maybe it just didn't work. Maybe it was too late. Maybe it was.
Yeah, I mean, it works in the lab, so it's like you assume like it works in the KPC model, but I agree. And this is how could they have determined if it actually did what they think it did? How do you think they could have determined?
Well, what about scans?
Okay, but you want to know if it actually broke down the hyaluronan matrix, right?
Yeah.
So how could you determine that beyond scans?
Could you give, I don't know, some sort of dye and then see if we put in, I mean, it's hard work to add a cell. I don't know, these are people, so it's very
Right. Yeah, in the lab you certainly could. You could do like intravital imaging and that kind of thing, but people don't appreciate intravital imaging. They tend to not like their organs to be stained or anything. So I think one way you could do it is like a monotherapy lead-in with a biopsy. And it's where you give the like hyaluronidase and then 8 days later you do a biopsy. So this is similar to like Brent and Hope's study in Key Largo, which is where they gave an intro pembro dose to patients with gastroesophageal tumors and then biopsied on a day and you could see changes. So it's like, this is a really good point because like you better know how your drug works. Like if you're gonna put it into people, like there's really no excuse for it, just for saying like, oh, so why didn't they do that in phase 1 or phase 2 and then say, oh, because you could have been right, you could have saved that company millions of dollars. To save companies millions of dollars. That's what we're all in it for.
I just think through out a good idea.
Yeah, that's good. But I think that's a good article to show like how that kind of biologically, sound idea is supposed to be paying out. All right, which one's next?
Dude, epicadostat.
Let's do epicadostat. That's a whole bag of worms. Who had epicadostat? Who wants to talk about it? Somebody. Dan's gonna call names. He's cruel like that.
Actually, yeah.
See, I didn't put that in. That's all him.
Oh yeah, I just went in alphabetical order, so I don't know who ended up on what list.
It's like all first years of mine.
Right last. Yeah, so we talked a little bit about epicadostat and its mechanism of action of being an IDO1 inhibitor. Well, what was the big caveat about IDO1 and its duplicative enzyme? Remember it, so it converts tryptophan to kynurenine,
Yeah.
right? And we know that tryptophan is a good thing, kynurenine is a bad thing. So theoretically, if you inhibit IDO1, good thing. But it has basically a duplicative enzyme called TDO. And so if you block IDO1, theoretically, at least based on the results of this, because you're really just shunting all of the enzymatic activity over to TDO.
So you got IDO1, you got TDO, you've got IL-4I1. So these are all alternative mechanisms of tryptophan degradation. And this one, admittedly, it was like one of these articles that you could have done like the deepest dive in reading it, because there's so many problems with this trial. I was actually there when they presented this phase 3, and there were gasps in the audience. People were shocked that it was negative after the phase 2. I mean, really, like it was like, oh, this is it. They're going to replace ipilimumab with epicadostat. So I guess these redundant enzymes are one. It's also a redundant pathway because it's stimulated by interferon gamma. What else is stimulated by interferon gamma? Oh, well, PD-L1. That's right. Then other things that you could have gotten from like literature review of that article is that IDO1 is a transcription factor, so inhibiting its activity does nothing to other things that it does for the cell. And that was like recently discovered, and I'm like, cool, sure. And then if you inhibit Wnt ligands, we have found that this is more effective than epicadostat, and it is not due to TDO or IL-4I1, because we looked at that as well. But it actually sustains the expression of IDO1 when you stimulate dendritic cells with Wnt ligands. So they are able to overcome the inhibition. But to your point about drug design before, and like how could they have determined if their drug's working? They never did. They didn't even measure like serum kynurenine and tryptophan or any of that kind of stuff, or do on-treatment biopsies. There's no biomarker. So unlike the hyaluronan study, or at least they had a biomarker, they had something they could have looked at the delta for, right? But they didn't, and we can say what we want about that. But in this, it's inexcusable. They didn't select high kynurenine tumors. They just went big. They're like, let's replace ipilimumab, and it didn't work. So epicadostat has failed in several of their tumor types. And I think there's probably multiple mechanisms of that. If you want to do some more reading on it, I'm happy to talk about it. There's some reviews up there too. So. All right, we'll knock out these last two and we'll release it and switch to anything else. First one.
So tiragolumab is a TIGIT, blockchain TIGIT is another immune checkpoint marker. I sort of think about it as similar to CTLA-4, it's like a separate compartment to PD-1, PD-L1. So Tezo, PD-L1, and they, this is a phase 3 coming off the heels of a phase 2 that led to emergency approval in EGFR mutated non-small cell lung cancer because of improved overall response rates. And so this trial is failed, but not without, some non-statistically significant benefit. But so it's a combination of anti-TIGIT and anti-PD-L1 compared to PD-L1 inhibition alone in PD-L1 high non-small cell lung cancer. And it failed to show statistically significant improvement in overall survival or PFS, but did have some trend in both categories.
What do you think they could have done better?
So it's interesting to me that the patient population selection is different than the phase 2, and so I wonder if there might be some collection of biomarkers or tumor variables or even mutations that would lead to a clearer benefit, though it's hard to, there are good inhibitors for EGFR and ALK, so it's hard to say, yeah, let's do this to them.
Yeah.
But yeah, I'm also not sure. I don't know enough about the mechanism of downstream TIGIT to say, is this truly distinct enough to be
Yeah.
feel it works.
I think that's great. I think your point about phase 2 population differing from phase 1 is extremely well taken, because that happens a lot. That did not happen in the epicadostat trial, because that was a suspicion. It's like, oh, they just had too many patients with liver mets, and that's why it wasn't the case. So that is something you should look at when you look at these trials. But there are alternate binding receptors to PVR, which is what TIGIT binds to. So blocking TIGIT is probably just insufficient, and there might be other better receptors to block. But I agree with you that they should have biomarker enriched. That's actually the biggest takeaway from that trial, so you got that one for sure. So, yeah.
They were agnostic to.
Yeah, and that's great. That's great. I'm not gonna sugarcoat it. That's great. Like when pharmaceutical companies are doing that, it's like you want the whole watermelon, not a slice. Okay, well, you might get nothing.
In this case.
Yeah. Bintrafusp.
Second, briefly, I didn't read the whole thing from cover to cover, but essentially the idea here is that in patients with oligometastatic colorectal cancer limited to the liver, you resect everything out, and then you look at ctDNA as a biomarker for if there's any residual disease. That was the patient population under study here. And the idea is, in MSS disease, we're going to try to eradicate it using this very interesting functional protein, essentially.
Mm-hmm.
So PD-L1 with what is ultimately a TGF-beta receptor trap. The idea that TGF-beta receptor signaling can exclude tumor-reactive T cells from the tumor microenvironment. So you're trying to really supercharge an immune response in an effort to fully eradicate and, I'm going to say, cure these patients of what was originally stage 4 cancer. The outcome is that they lost equipoise immediately, and the people that were treated with this, and it was, I think, just a single arm trial compared to historic controls, but the people that received treatment had ctDNA rapidly increase, and then that was followed by radiographic progression of disease that was significantly worse than the historical trial outcome. And some of these folks, and I didn't look at the specifics of it, but the thought was that there was TGF-beta3 that was upregulated, compensating, and ultimately leading to these worse outcomes. And their big key takeaway is we don't understand the biology like we thought we did. And that led to worse outcomes, significantly worse outcomes. A very small study, but run out of MD Anderson.
Yes, yeah, and the lead investigator, I will always add this caveat, absolutely 100% did the right thing. And didn't push on with his investigator. He could have done a selfish thing and pushed on with his investigator-initiated trial and said, oh, this is all recurrence. Nope, he stopped, he did the right thing, questioned the data. So that's the takeaway. What do you think they could have done better, other than like not giving people disease?
Right, yeah, well, not giving people worse cancer, it's number one. But, you
bad
know,
move.
So I am blissfully unaware of the context in which this was done in the preclinical setting and what they saw in mice. And then it kind of goes back to the point you kept on going back to, which is that mice and humans are different.
Yeah.
So I would imagine they probably had very promising preclinical data that then just the biology was fundamentally different in humans. And I don't know what other biomarkers they looked at besides just the ctDNA, and if like the people who didn't have such a bad disease have that TGF-beta receptor, or the TGF-beta3. And I think there was also cases where when they stopped the drug, then the tumor actually settled down a little bit as well.
Yeah, which that kind of speaks to probably like an epithelial proliferation driving mechanism, that it's like inhibiting the TGF-beta is pushing those tumor cells more towards like a mesenchymal to epithelial transition. And that was one of the other things I proposed. But you nailed it. I mean, it is a study that lacked sufficient rationale and had to be stopped early and actually caused patient harm. I think that's a good one to end on. It's like know how the immune system works, and maybe we'll drug it better. All right, thanks guys, appreciate it.
Thank you. Fantastic. Always a pleasure as well.
Same to you.